Okay. We're starting.
Thank you, Olive. My name's Megan, and I'm part of the Ionis study team.
And my name is Abby Collins, and I used to be in practice at Children's Hospital Colorado and took care of a couple of people living with Alexander's disease. And then I started working at Ionis, and I'm the project team lead for the Alexander disease trial.
Hi, I'm Amy Waldman from Children's Hospital Philadelphia. Why don't you guys come up?
Join the circle.
Join the circle. We're all gonna like.
Tracy, circle.
Um,
It's a small group, so this works.
yeah. So I'm a pediatric neurologist. I see though all ages. Come on in. We're having an intimate session today rather than doing the chair thing. And how I'm affected by Alexander disease, I'm affected when all of our patients have an issue or concern or a problem. And I take your stories home with me and want to make it a better place for everyone that's dealing with this disease.
So I am Francesco Gavazzi. I'm a scientist at the Children's Hospital of Philadelphia, and I'm relatively new to the Alexander disease world. What I would like to do is trying to understand what are the best ways to evaluate children appropriately to then understand if what is done by the very smart people on my left actually works effectively and can be demonstrated to any kind of level that can then approve the medication if it works.
My name is Carlos Dominguez Gonzalez, and I'm a genetic counselor at CHOP. And I mainly, my involvement with Alexander disease is helping first in the diagnostic process, so help people get a diagnosis of Alexander disease, and then help families understand the implications for everyone in the family, and cope with the diagnosis.
My name is Yumiko Kinoshita. I was diagnosed with the disease last September. I've had symptoms from about 5 years ago, the very first inkling of something was wrong. And I'm based in New York, New Jersey, and I've met with Dr. Waldman in CHOPS. And I'm glad to be here with folks. Thank you.
Hi, I'm Kaz Taguchi. I came from Tokyo, Japan, and I'm a entrepreneur of Astellas Pharma, and making a drug development platform for rare diseases.
I'm Rob Goldstein-Mahon. I'm with Ionis Pharma, and my relationship to Alexander disease started when I joined Ionis a couple of years ago and had the opportunity to join Abby's team and working on our drug development program for zilganersen. I've been working in rare disease for 20-something years, and I'm just really excited to learn and listen and learn from all of you today.
Hello, everyone. I'm Yukari Blumenthal. I'm a mother of Kuno, 6 years old, boy affected by Alexander disease. Beyond that, I used to be a urologist, not a neurologist, urologist. Confused. And I also work for Astellas Pharma, and we have been supporting early stage research and development for the cure of Alexander disease. Yeah. Nice to meet you.
Hi, everyone. Jason Child. I'm Olive's dad, and I have been in this community for the last 11 years, I believe it is. Happy to be here. A lot of familiar faces. My current passion and primary reason for being here today is to be recruiting and encouraging everybody that has been affected to provide their voice and their story. We are, as I'll talk a little bit more later to this session, later on, preparing for a voice of the patient submission to the FDA, which we need all of your voices and all of your help in doing. And so I am here hat in hand, as I mentioned to many of you a month ago, begging for your participation and at least giving me an opportunity to tell you why I think it's worthwhile. And it's very nice to meet some of the people I haven't met yet.
Hi, I'm Ylenia Vaia. I'm a pediatric neurology at Milan's Children's Hospital. My journey with Alexander disease started relatively recently, like 3 years ago, by chance during my residency school, and then became kind of a purpose as our community grew bigger and the trial kept going on, and then it became my main subject of study as a PhD student, and it's my it was my residency thesis theme and will be my PhD thesis objective.
I'll let you go. Hi, my name is Olive, for those that know me. And I am one of those patients that has Alexander's disease and goes to CHOP and is.
What's your favorite thing to do?
I like bowling and I can do most things. Thank you. Bye.
Thank you. I love your smile while you are eating lunch. So happy smile. I'm so happy to see your smile. Yeah, please. Yeah, take it easy. I'm happy to see your stories. Yeah. And I'm Sayaka. I'm also Astellas employee, and my responsibility is to improve the access to health located in Chicago. And I'm also working with Yukari Kuni about the development research for Alexander disease. It's nice to meet you. Thank you.
Hi, I'm Stephanie Curtis. Phil and I have an 8-year-old daughter. She was just newly diagnosed a couple months ago. So we are just getting caught up to speed with everything in this world. So we are new and we are open to everything.
Phil Curtis, and I had a good smiling picture of my daughter. There we go. And so we're in this community since April 18th. And to come, honestly, to get the diagnosis and to feel so alone, and then just to get the support from the community, which was obviously a couple weeks, is pretty touching. And, we're looking forward to do what we can. And the focus, as parents is just to give our daughter the healthiest, normal, and happiest life that we can, and whatever obstacles we just want to overcome. That's the primary focus.
My name's Ben. I'm a researcher. I run a research lab at Case Western in Cleveland, Ohio. I study astrocytes. That's the focus of my lab is astrocytes and when they sort of go rogue and do damage, as I'm sure most of you guys here know. And my connection to Alexander disease started when Thomas Wagner actually reached out to me after a paper I published, and I was aware of the disease, but sort of on the periphery, and have chatted with him a few times, and I'm here to learn about the community and get involved.
Hi, I'm Tracy Hageman. I'm a researcher at the University of Wisconsin in Madison, so it was a quick, easy drive to come down and visit with everybody, and I think it was a good choice to do. So we've worked with Ionis on some of the initial studies, developing the ASOs and rodent models, and they've obviously continued quite a bit with that, and hopefully successfully. But I've worked with algae messing for about 25 years, and so over the years we've developed rodent models, and I think those have been put to good use, but it's always good to actually get out of the lab and meet people, and so I enjoy these experiences, and I'm happy to be here and learn a little bit more about the human experience, you know, get out of the lab a little bit. So it's nice to meet everybody. Thank you. Is anybody left out here?
Hello, nice to meet you. My name is Kuni Ito. I'm also working at Astellas. I do various work at Astellas, doing some research and also working together with the colleagues. I also working at the university in Japan to do some social science work, to propose some new regulatory framework to enhance the rare disease type drugs. So that's another phase I have. I'm very much interested in, and also happy to join this community and talk together. Thank you very much.
I don't know if we still have our friend. Do we know who's online? Do we know how? Yeah, we have 3 people online. 3 people online?
Yeah, that's why we keep using the microphone, just to be sensitive to them so they can pick up on it.
Loud enough for the people online, or do we need to be louder? Or?
Yeah, you should be good. Just make sure you keep the microphone up to your mouth.
Okay. Do we want the people online to introduce themselves? Are they live?
Yeah, everybody online, would you like to unmute yourself and introduce yourself? Or do you want me to just read your names? Up to you guys.
I'll go. My name is Tom Cancellieri, and my son Levi was diagnosed August 27th of 2021 when he was born, 32 days later. He's been a patient of Dr. Waldman since October of 2021. He's doing pretty good. We were told he wasn't going to make it to B2, and he'll celebrate his 4th birthday next month.
He'll tell you I was not the one that gave him that. Just saying. I was not the one that said. They came to me and I said, no, we got to throw that out, right?
You guys want to pass around 2 mics?
Sure. Yeah, Olive, you want to go? Olive will grab it. Oh,
or runner.
Hey, I don't know if you can hear me. I'm Taya, and I have Brandon here. Our bub is 6 months old, and she was just diagnosed on Tuesday. So we are here kind of seeking information, hoping that there's a trial drug or something that we can help with her.
Wow, I think you have silenced us, many of us in the room. We want to welcome you to our community. Of course, it's not anything that anybody wants to join, but you have a lot of people here that are going to help you, support you through this journey.
Thank you.
Was there one more person online?
Sorry, it's Emily. I have bad background noise, so I apologize. It sounds like I'm at a rave. I'm just sitting in a coffee shop. Are you guys able to hear me now?
Oh, we can hear you.
And there is no background noise, so you're protected.
Oh, I'm glad. Okay, good to hear. Well, nice to see everyone. So I'm joining remotely. I'm Emily. I'm mom of Atlas, and we're a couple years past diagnosis. We were a patient of Dr. Collins, so nice to see you. So glad you're still involved in the community. And now Dr. Waldman's patient. And I'm a new member of the AxD board as well. So glad to be part of many things.
All right. Well, welcome everybody. So, you know, I think these kind of sessions are always interesting, right? I think last year we had a summit in Philadelphia and we had over 100 people join, and people from all over the world, and it was a remarkable event. And you know, we wanted to try and recreate that on a smaller level, kind of tapping into the ULF. I should introduce myself. My name's Jenny. My name's Jenny Pierce, and I feel like I know most of you in the room. I am Elise's mom. She was diagnosed back on November 20th, 2014. She is now about to turn 14 years old. And I was explaining, as some of the moms were saying, you know, missing the kids and being hard. My kids don't care at this point. They're like, all right, mom's gone. We're good. So I first became involved. I met Dr. Waldman, you know, early 2015. I think Elise was probably one of your first Alexander patients. And in that room, we just kind of connected in a way that was not based in anything like sane. But it was just like a connection that we formed, and we said we wanted to do something to change. And now, how many patients over the world have you seen?
Over 100. We stopped counting. Oh, we've seen over 100 patients clinically and for research at CHOP.
Yeah. So, you know, Dr. Waldman, her team, and other leukodystrophy parents really inspired me to go back and do something, which was to start a nonprofit. Which was called Elise's Corner. And we've done a lot of work over the years raising money to help drive research in the right direction, just so that we would see a day like today. So, you know, I'm here to say, I know we have several newly diagnosed people that being diagnosed 10 years ago, you know, looks so much different than being diagnosed today. And I'm hopeful that we're able to share some information. I'm not pretending like it's easy. It's not. But I think that it's really important that we draw on the hope. And I think where we are today in terms of the hope of what's happening in the community, as well as just sitting here in this room, and AxD and who we are, you know, and where we're going, I think that there's a lot of things that we need to be hopeful for, because we're a strong community and we're committed to growing even more as we go. So, Olive, you can switch that. All right, this is a disclosure statement. The ULF makes us say it. I'm not going to read it, but there it is. Okay, move on. So this just kind of goes through sort of the things that we were planning on talking through regarding this mini symposium here. There's a lot going on the screen, but really, you know, we have all the key people here. We did the introductions. Going to give a brief AxD update. You know, and especially we want this to be designed to be as informal as possible. So please, people on the phone, I mean, just jump in, ask questions, do whatever. Dr. Waldman's going to talk a little bit about clinical research at CHOP, just what's going on there. Again, not too medically focused, but in a way that we could all understand. Dr. Vaia, you're going to talk a little bit about the updates that you guys are making. Yukari, who, interesting, a mom of a kiddo with Alexander disease, as well as a scientist, she's going to provide some updates that's going on at UMass. We'll take a little bit of a break, and then I think the most impactful is really just that open dialogue. And I don't want any of the scientists or researchers or anyone to feel like you're not part of this, but, you know, just connecting and really talking about what matters to us, and, you know, talking about our journey. And it's isolating and it feels so alone, and whatever we can do to help support one another and help people feel empowered, so that we can come out of this being the best possible organization. I mean, I say this all the time, like, I would put us up against any other rare disease community, and I stand by, partially because I'm competitive, but also too, I like a challenge. But I think that we really have just assembled, I think that we're all here for a reason, and I think we're headed in a really, really good direction. So more to come on that. And then finally, Jason's going to be talking about our journey going before the FDA with our patient-focused, what is it? Patient-focused disease discussion, drug discussion.
Going before the FDA.
Yeah, but you're not allowed to say the FDA. But anyway, yeah. So talk externally led. Yeah. So he's going to give some updates, and then yeah, absolutely. Yeah, Olive's going to jump in wherever possible. So very fluid, very informal. So again, I mean, if there's any questions or anything, you know, I'm looking to you guys as the patients, especially newly diagnosed, whatever questions you have, just please jump in, because this is about you guys. All right. Next, please. Next, please. All right. So before we kick things off, and actually we're going to go to the video next, if you don't mind pulling that up. Clayton, back of the room. Can we pull up the video? Oh, perfect. Thank you. Wanted to share this video. It was something that aired. We actually had the opportunity to have Alexander disease showcased in the national spotlight. Thomas, who is not able to attend today, one of our AxD board members, he was featured on the Today Show. I think they did an 11-minute long session on him and his journey and his commitment to helping save his son Max. So let's go ahead and watch that quickly.
Hope is a funny thing. If you didn't have any, you wouldn't do any of this. Planning a family is one thing, getting pregnant is a whole other situation, so it wasn't an easy journey for us at all. So when we finally got the news, you almost don't want to dare believe it. Max was born in June 2022, and we weren't the youngest anymore when we had him, so we had a fairly established just the two of us routine. And obviously then it was the three of us. Max was always incredibly friendly. He very quickly started cooing and oohing and eyeing and sort of had a lot of things to say. December, I was going to start my dad leave, but unfortunately, that's, I guess life had some other plans, because that's when when Max had his first seizure. Stephanie woke me up in the middle of the night, 4 a.m., in panic, and was like, Thomas, I think Max has a seizure. And yeah, he clearly looked like he was having a seizure. So we rushed to the hospital. It was full emergency room. They immediately did all kinds of tests. Did he fall? Is it an infection? Is he ill? At which point do you go like, okay, something is absolutely not right. When the genetic test results came back, Max has Alexander disease. I had no idea what Alexander disease was. Typical life expectancy is 5 to 10 years. There's no treatment, there's no cure, there's nothing we can do. And yeah, I fell to the floor. You mean you have all of the emotions. You're angry. It's unfair. What has he done? Like, he's just 7 months old. I mean, I love him so much. And to just accept the fact that, you know, we'll just wait and see and do nothing is, that is not an option. There's always something you can do. The first thing you do is search, like, what is Alexander disease? Because you have no idea. Okay, Alexander disease is caused by a mutation of the GFAP gene, which makes a GFAP protein in astrocytes. And then you're like, okay, what are astrocytes? So you start hearing all of these words, like myelin and oligodendrocytes. Like, I don't even understand the language. When Gemini came out, the really lovely thing is that it can read PDFs. So it can help me analyze and understand scientific papers that are incredibly dense, could be 20, 30 pages of technical language. And I can go, just summarize this for me, please, and help me understand. Then I get a very helpful top-line summary of what this talks about. I think that's where I started to get a bit of a sense of what was going on. I understood, I guess, that there are some parallels between Alexander disease and Alzheimer's. And there's not that many people working on Alexander disease, but there is an entire army of pharmaceutical companies, research scientists, and everyone in between working on trying to fix Alzheimer's. You realize that suddenly there is a bunch of people out there who work on things that they themselves don't know might be really relevant to Alexander disease. So I can literally take all of these papers and I asked Gemini, can you help me understand this paper on Alzheimer's? Thank you. Can you help me summarize this paper about Alexander disease? Can you bring the two things together, please? How is this relevant in that? Oh, okay, this might be relevant. Hey Gemini, can you help me write an email to the author of this paper outlining how their innovation might be relevant in Alexander disease? And it's like, sure, can. I literally send an email on Friday late afternoon. Monday morning, I get an email back from this professor in New York.
That email really caught my attention. I have to admit, we've never heard of Alexander disease before. You know, I emailed him back saying, like, I think this could potentially be something that we could test.
And now she is working on Alexander disease. They're literally in the process of arranging the experiments. That is mind-blowing to me. Probably send 1 or 2 of these emails a week. Scientists are mostly responding.
I really thought that I was speaking to another scientist. I'm talking about like someone who has a background in artificial intelligence and algorithms. The email, if you read it, it was like your algorithm can be used to identify something that interacts with the molecule that causes Alexander disease, and detailed why.
He asked the question, what is Alexander disease? But then he took the answer and started asking why. Why are the astrocytes affected? Or why is there blood-brain barrier breakdown? He started to apply that and say, well, what if we approached it in this way?
The leading scientist in Alexander disease, who I've pestered quite a lot, he said to me, like, there is no chance you have no background in biology. I was like, no, I have absolutely no formal training in biology. Whereas that Gemini has been incredibly helpful. I genuinely am not making this up. Alexander disease falls under the category of ultra-rare diseases. There's thousands of rare diseases that you have never heard of, but that still adds up to hundreds of millions of people around the world who are affected by a rare disease, who are being told, sorry, there's nothing much we can do about this. Yes, they are incurable at the moment, but that also means things just haven't been tried. If you're being told that you have zero chance, then every scientist working on a thing that might be relevant increases the odds above zero. A lot of it is about buying time. If you can slow some of these things down, that gives more time to the scientists who are working on a cure. At the end of the day, Max is still a hilarious, almost 3-year-old little man who, has a little personality and wants to have fun with Dad. So at a very, very, very basic level, we hope for him to, to live. Stephanie and I said we were not gonna give up, and we're gonna do what we can and what is in our power to do. When we said that, we didn't necessarily think about rallying scientists to help figure things out, and there's probably 5, 6, 7 research groups that have started working on Alexander disease because I sent them an email.
It has made us, take all this new path that we wouldn't have taken otherwise, right? And I think that's why what he did is so important, to actually improve and increase the number of researchers that work on rare diseases, like making them aware that the work they are doing is useful and applicable to other settings.
Thomas has kind of created the blueprint saying, there are people that are willing to work on new problems that may not even realize that their technology can work for that, because we just don't have that bandwidth to see beyond our siloed perspective. They make that connection. By doing that multiple times, we probably will solve problems that otherwise would just go untouched.
And the fact that Thomas is so aggressively pursuing these changes to the field, not even knowing whether it could help Max, it is the most amazing, selfless thing that anybody can ever do.
I don't think we're gonna stop. Why would we? We might succeed, we might not succeed, who knows? I mean, it's life, but he deserves a shot. I'm Thomas. I'm not a scientist, I'm not a doctor. I'm just a dad.
Which makes me be so confident in saying I'd put us up against anyone else, because he certainly has a way of I guess not backing down and not just taking no for an answer. So really, really proud to have him on his team. I will say, okay, so what I wanted to do is I wanted to share our 2025, our End AxD board. And again, I mean, we've really been focused. Jeez Louise, I'm gonna fall. I'm gonna step out of everyone's way. But when we started to build this organization out, we said it has to be more than just research. It has to be more than just advocacy. It has to be some of the touchy-feely stuff. It has to be the patient support, the education. It has to be about fundraising. So I feel like we've really compiled almost a dream team of people. that are really, really passionate about the different areas that we're working on actively. So rather than our pictures, we decided to put up the pictures of our loved ones affected by the disease. Again, my name's Jenny. I'm serving this year as the president for AxD. That's my daughter, Elise. I will be working on primarily patient advocacy, with the support of Jason too, as we start to think about what it looks like and what our messaging looks like as we go to the FDA. Jason Child, we brought him on, in terms of just having that strategic oversight, making sure that we're not missing anything, that we're not becoming too siloed, that we're not just fixating on one thing without, kind of considering the bigger picture. And that's a picture of your dad, right, Olive? No, me. Oh gosh, yeah, that's you. Sorry. Oh, okay, that sounds like a story. Thomas Wagner, who you just saw on the video, that is his sweet little boy Max, and Thomas wanted to be here, but he and his wife, they're expecting their second child. Yeah. I thought. Ooh. You guys will be getting emails. Emails are coming out from Thomas shortly announcing it. No, he had wanted to come, but he and his wife are taking a baby moon. So yeah, so they're doing that. So couldn't make it. Yukari, she is actually, a mom. She is also not only a board member, she's also a member of our scientific advisory committee. So one of the big things that we wanted to accomplish to really make ourselves kind of be legitimate is establish a scientific advisory committee to make sure that, anything that we're giving money to as an organization, we're making sure that we're doing the due diligence and making sure that we're really, really vetting everything properly, and that the money that's going out is definitely in line with our vision. And then Gil down below there, and I want to point out too, as I'm kind of going through this too, I mean, I'm from Cleveland, Ohio, Jason's from Toronto, Thomas is now in Northern California, Yukari's from Japan, Gil, he lends his hand to research support. He's based out of London. He actually also is a board member of AxTLC, which is a European-based leukodystrophy support organization. Tyler is based out of Seattle, and Tyler was actually the one who was kind of coordinating this entire meeting. And I'll say this because I know we all care. Tyler actually went to the ER 2 nights ago with some severe pain and just found out he has cancer. So I don't know. I mean, definitely a journey ahead for him. I know that it's affected our board pretty significantly. Not only is he really the leader of his family and all that, just such a fighter and such a good human to Eli and Jill and his other son, but now he's got a battle of his own too. So we're all kind of stepping in and trying to wing this presentation as we go. To our fundraising, because again, you can have all the research you want, you can have all the advocacy you want, you can do all of this till you're blue in the face. We need to have money coming in, and we need to make sure that we're generating money from different sources in order to be able to follow the projects that we're passionate about. So we just brought on board Emily, that's her son Atlas there. Emily's on the phone as well. And then Deb Hughes. Deb was here. She had to leave. She had a family emergency too. But that is her son Daniel. And the heart on his picture, we lost him. The community lost him, I want to say it was late last year that Daniel passed. So definitely a brave kiddo, and he fought really hard. And then last but not least, we have Laura Ledbetter. She is really passionate, and she's way better at this than I am, and probably many of us, at just really finding out a way to connect with families and show them the love and the support. She is notorious for sending out the cards and birthday bears and all that kind of stuff that just makes people feel like they're loved and they're seen and they're supported. So that is our board. We are small, we are mighty, and we're all probably being pulled in about 10,000 different directions, but there we are. We say we're not perfect. We're aiming at crawling before we can walk. I think we're getting there. We're getting pretty close, and we're going to fail. I'm sure we do every day, but we're failing moving forward, and we're failing together. And we're really focused on just getting better day in, day out. So, all right.
And thank you to Kristen for all of
Oh,
that
yeah.
she's been doing.
And we have our unsung hero, Kristen, who is not connected to this crazy world, but we dragged her in. She's the one who kind of micromanages all of us and makes sure that we're reading presentations and editing. And she has a whole standard of code of conduct, a mission, our plan, our this or that. She's responsible for all of our social media, everything, all the things that we just don't have time for, that she's really picked up the slack and done. So, Olive? Okay, I think you abandoned the clicker. Alrighty, so with that being said, we can go ahead and switch over. I know Dr. Waldman had some things that she wanted to cover. I think there's a separate presentation for Dr. Waldman, so we'll go ahead and hand it over.
Clicker.
And again, jump in with any questions or if anything that doesn't make sense. This is for you guys.
All right. Well, it's great to see everyone and happy to walk everyone through the last decade of work that has come out of CHOP and where we're going from here. There were some emails that were perhaps a little confusing for some of the community. So want to make sure that everyone understands where the natural history study is at CHOP and where we're going. So as soon as the slides come up, then what was your name? Waldman, Amy Waldman. So as we're pulling this up, so first of all, to the new family on the phone, you did hear a little bit about life expectancy. Ignore it. We're going to be planning lots of birthdays and doing everything that we can to support your family. But I want to take us back 10 years ago. We decided that we needed to collect natural history data, and this is really your data, the community. So we have a lot of publications in the works, and I just want to walk you through some of them. Oh, here are my disclosures. And I'm going to begin with thanking all of the families. I love this quote: "When children inspire science, extraordinary discoveries happen." And it's not just children, it's the adults as well. We learn from all of you and would not have been able to do half of what we've done. And I would not have learned half of what I've learned if it wasn't for your belief in me and coming to CHOP and letting me be part of your journey. Okay, so I'm going to just, in my couple minutes today, I want to go through where we are with the natural history study and some key takeaways that we've learned. We're going to go over some changes, right? We've been doing this now for 10 years. You've come to see us at CHOP. You've gone through the physical therapy and the occupational therapy, and you've done all these things, but where are we going to go from here? And then the importance of community participation. Okay, so I'm going to start with our publications, and I'm just going to go through a couple of them. So we have published a paper on developmental milestones. This paper, I think, was so important because I think a lot of families are told you can't when you get the diagnosis, or you won't be able to. And so we wanted to counter some of that with what are the realities of the developmental milestones? When do patients learn to walk? How many learn to walk? So we published this paper. I will say this is centered on what I call the cerebral phenotype. Other people call it type 1 or the infantile. It is not focused on the neonatal, and it's not focused on adults. And the reason why, the neonatal phenotype is a little bit different. And you heard the Cancellieri family on the phone. They were given the prognosis of the neonatal phenotype, which I didn't think that their son had. I'm sorry. I hope you don't mind me sharing all of that. But this is the cerebral phenotype. It's also not the adult or the intermediate phenotype because they usually have normal or near normal developmental milestones. So if I included those phenotypes, it would have biased the sample. It would have made it look different. So this is really just focusing on the patients that present with seizures or developmental delay. So we looked at some time to events, like when do you develop things? And I'm not going to cover everything that's in this paper. And we've reviewed patients' developmental histories. So a couple of key things. One is that gross motor delay is the earliest symptom onset that we're seeing in the cerebral phenotype, followed later by seizures, and even later than that, the macrocephaly. And that's demonstrated here in the blue and the green and the pink colors. You know, I think part of the delay in the diagnostic journey is that people are waiting for you to have all three of these symptoms. And so the importance of this paper is saying these symptoms don't come at the same time. If you're waiting for all three, then you're not going to be able to make a correct diagnosis. Yeah. Enlarged head size. So a big head. So we wanted the physicians to know that these things, physicians look for patterns. And so they look for the pattern of all three of these symptoms. And if they don't have an enlarged head size, people don't always consider Alexander disease. But in our juveniles and adult patients, many of them have a normal head size. So trying to make sure that we explained the differences in some of the symptoms. We also put, this is a busy table, but it goes through when we expect developmental milestones to hit. So in that first column, the number who achieved the milestone, you can see 100% achieved head control, 98% rolled over, 96% sit without support. And it decreases a little bit. 67% of our patients walked independently. Another 20% walked with assistance. But if you look over to the right, the mean age of being able to do that was almost 2 years old to be able to walk independently, and just short of 4 to walk with assistance. And on the bottom, you have combining words. You know, we think about combining words in a unaffected population around age 2. This was closer to age 3. So the purpose of this table was really to say to physicians, do not give up on these kids. They're going to be late, but they might get there, and we need to be aggressive with physical therapy and occupational therapy and all the therapies in order to make sure they achieve these milestones. Okay. Another paper that we've published is about the use of anesthesia. So for many of the younger children, they go through anesthesia to get an MRI or a lumbar puncture. So we published this paper on the safety of anesthesia at CHOP. We did not have serious complications from anesthesia. I will say that one of the parents really made me think twice about this paper after we published it. And I want to make sure that I share that just because we are saying that anesthesia is generally safe does not mean that Alexander disease patients don't have sometimes side effects from anesthesia. So in general, at CHOP, because this was only at CHOP, when you had an MRI, so that's anesthesia for about an hour, or an LP, which is anesthesia for about 20 to 30 minutes, or sometimes combined an hour and a half, those short courses of anesthesia has generally been safe. We've not seen patients lose milestones after anesthesia. But it is true that patients in our community have had anesthesia and have had setbacks. So again, I want to just make a statement that not all of research results apply to every single patient. And I was giving the example when we were sitting in the hallway talking about mammograms, right? Many of us know that women, once you get to be a certain age, have to start undergoing mammograms. But that's never to say that women weren't diagnosed at 35 with breast cancer or before they would get their first mammogram. And maybe it's okay for the general population to start at 40 or 45 or 50, but there are always going to be women that are exceptions to those rules and have breast cancer earlier. So research, I want people to think about is more of a generalization sometimes and not always a holy grail for an individual patient. So just that caveat there. This table in this paper goes through all of the things that could go wrong when you get anesthesia, right? So just pointing out a few things. If you have seizures, right, the concern on the right is that maybe there are breakthrough seizures on induction or emergence through anesthesia. That is rare, and we didn't see it in our cohort, but this is the reason we did this paper, because we were worried about these things. A lot of our patients have problems with swallowing. So coming in and out of anesthesia, the burden of oral secretions and drooling, like they could aspirate, right? These are all the theoretical risks that could go wrong. And so these were the things that we were looking at. But I think this is helpful for people to be able to show their anesthesiologist to say these are the things that we worry about in this condition.
So Amy, when you talk about anesthesia and possible breakthrough seizures, when would you expect those breakthrough seizures to take place? Coming out of anesthesia?
Within 12 hours, within a couple of hours.
Couple days?
Not usually.
Couple days?
Weigh in. But this is a perfect example where I'm saying in general, no. There's always exceptions, but I think that if we were expecting, to the other neurologists and stuff in the room, I think that we're basically emerging from anesthesia, because when you're under anesthesia, and jump in if you want to say something differently. When you're under anesthesia, the whole point is to go through different your like sleep cycles in the brain, right? To put you, we don't like to use the word put to sleep under anesthesia, but you're when you go in and out of those different stages of sleep, that's when you can trigger seizures.
Oh,
So it really is about the emergence from anesthesia. Do you want to weigh in? No. Olive, do you have a question?
Like, when do you typically start to get this?
To get Alexander disease or the anesthesia?
Anesthesia.
Oh, when you get anesthesia or sedation, people use different words. When we want you to relax for an MRI, like the pictures of your brain, or for a spinal tap, right? If they want to put a needle in your back. A lot of people get anesthesia for those procedures. Okay. It's a good question. Okay, this is a paper that just came out. It is about increased opening pressure. So we started doing lumbar punctures, spinal taps, in our natural history cohort because we wanted to collect spinal fluid to test GFAP. So we wanted to test the protein in the spinal fluid, so we started doing these lumbar punctures. And as any good neurologist, part of the procedure is to measure opening pressure. To be honest, we weren't really thinking anything when we started this. We were just doing what we're supposed to do as a neurologist. And we started to notice that some people had increased pressure, which we weren't expecting. Now, when you have increased opening pressure, people worry that you have a disease called intracranial hypertension or increased intracranial pressure. So the opening pressure is a sign that you might have this increased pressure in your brain. And people actually associate the two and kind of interchange the two. And I want to be very careful about not using elevated opening pressure as a substitute for intracranial pressure. Increased intracranial pressure, you typically, in someone who's suffering from that, they have vision changes, they have blurred discs in the back of their eye, and some MRI changes. And they can have headaches, they can have some other symptoms. So in my mind, I use the term increased intracranial pressure in someone who is symptomatic, and an elevated opening pressure is something that we can measure to verify that that's the problem contributing to the symptoms that the patient's having. Interestingly, in this paper, we looked for clinical symptoms, we did the eye exams, and we did the MRI to try to look to see, is the opening pressure clinically relevant? So here's what we found, and this is what we just published. The normal range of opening pressure is less than 28. That's the 90th percentile in children. 25, quite honestly, is also a number that could be used, but that's for people who are non-obese, which a lot of our patients are not, but also non-sedated. So we chose 28 to be a little bit more conservative. So 28 is the red line in the figure to the right. We also maxed everyone at 36. We did not use extension tubing. So there's a couple of those points at the top that are measuring 36. We didn't go above that. So we just assigned 36 if the opening pressure was going, out of the top of the tube. We really did not see symptoms attributed to increased intracranial pressure except in 2 patients, and I don't know how well it's projecting. This patient here, we did the spinal tap. We noticed that the pressure was high, and we asked the family, does she have a headache? Is there anything going on? And they said no. But they said, oh, well, she does have this one symptom where she rubs the back of her head. We think it's just an OCD, that she just is kind of obsessed with rubbing the back of her head. And we said, I don't know. I think that maybe she's trying to tell us that she has a headache. So we started her on treatment, right? There's only these 2 patients that we put on treatment. We started her on treatment for increased pressure. And you can see when we repeated the spinal tap, it came down, kept her on it for many years, to the point where I was arguing that maybe we didn't need. Thank you. Maybe we didn't. That's perfect. Maybe we didn't need to still be on it when her pressure was down here and nice and low. And so we took her off the treatment, and then her pressure started to slowly increase. And when it was high again, she started to have symptoms again, and we ended up restarting the treatment. So this was a patient who had symptoms that we think were attributed to increased intracranial pressure. The earlier aged patient right here, it wasn't clear that symptoms that she was having were really related to increased intracranial pressure. Thank you. But she had a lot of vomiting. Many of you have experienced that vomiting is common in this disease. There were some other things going on. We decided to treat her for the intracranial pressure. We put her on the acetazolamide. Her vomiting did not improve. Her vomiting did improve when we did other things, but her vomiting didn't respond. So I don't think her vomiting was related to intracranial pressure. But these were two examples where we did treat, and so we wanted to show that the pressure did come down with treatment. So none of the patients, interestingly, had an abnormal eye exam. So when you have increased intracranial pressure, a lot of times we will send you, or increased opening pressure, we'll send you to an eye doctor. He'll look in the back of the eye, and if he sees that the back of the eye is blurred, that implies that the brain or something's pushing against the eyeballs, and he will start a treatment. Interestingly, he does not follow lumbar punctures. He only follows the vision and the back of the eye, and when that's better, you come off treatment. And so we've had a lot of discussions because with a normal eye exam, there's nothing clinically other than symptoms for us to go on and for us to treat. So it's been a little bit of an interesting journey to say, does everyone who has an elevated opening pressure need to be treated? We did see some findings of increased intracranial pressure on MRI in the group that had opening pressure, but we really need additional studies to understand, do patients need to be treated? What is happening? I didn't put in here the mechanism. As a doctor, I always want to know, why is something happening, right? Why do we have increased opening pressure? We do know that there are astrocytes, and Ben can probably tell you a little bit more. There's astrocytes around, and Tracy, there's astrocytes around the areas of the brain that control the fluid that surrounds the brain. So the theory that we have is those specific astrocytes are releasing GFAP, or there's a lot of GFAP that's clogging up the drainage system in the brain. And that's why you get these increased opening pressure, because there's so much protein that's draining the natural circulation, right? Your body is constantly producing new fluid, and we're measuring the opening pressure in that fluid. So that's our theory behind this, and why this is happening. The other interesting thing that I will note is that it's really in the patients under age 10, right? So when you get to over age 10, the likelihood of having this increased opening pressure decreases. So you see it a lot in the younger individuals, and it's not something I worry about as much in our older patients that have Alexander disease. Why? Because that's yeah.
I just confirm, is that hydrocephalus then?
That's a great question. Hydrocephalus is not the same thing. So you can have increased opening pressure in hydrocephalus, so that's true. But hydrocephalus to me implies a couple of things. One, there is a blockage, a physical blockage, that's obstructive hydrocephalus, right? There's something physical like a mass or something that is blocking the drainage of the brain, and then you get a backup of the fluid into the ventricles, right? And ventriculomegaly does not necessarily mean hydrocephalus. So all of these terms are different and not interchangeable. There's also, just to make it more complicating, there's something called communicating hydrocephalus, where there is no physical blockage, but the difference there too is that you get this increased pressure back up into the ventricles. So we have not proven, there are cases in Alexander disease of hydrocephalus for sure, but I don't think that every case of increased opening pressure is related to hydrocephalus. That's a great question. Did that answer it? And in the MRIs, we have MRIs from these patients, and we did not have hydrocephalus on those MRIs, or the ventriculomegaly associated with hydrocephalus on those MRIs. So this phenomenon is happening outside of hydrocephalus. To answer the question about why is this in the younger kids versus the older kids, so spoiler alert, we have our paper coming out on GFAP. We're going to be submitting that later this summer. There are much higher levels of GFAP in the spinal fluid of the younger patients compared to the adults. I did not put that in here because Albie taught me that I should not be talking publicly about stuff until it's peer reviewed and published. So I don't have all the figures in there, but I do think that there's an increase in GFAP in patients who are also under age 10, that the GFAP levels are just much higher in that patient population, and then it decreases over time. And I think part of that is because of the amount of brain that is involved in the children versus the adults. So in the younger patients, you have a lot of disease in the supratentorium of the brain, like the frontal part of the brain and the brainstem, whereas in our adult patients, the frontal part of the brain is spared and you have disease in the brainstem, but I don't think there's as much GFAP because so much less real estate is involved in the older patients compared to the younger ones. So I think that's part of it. Yeah, go ahead.
Yeah, but they were talking, so I was just. Is it okay? Can I ask? Yeah. So my question was, I was wondering, it's also because the amount of astrocytes genesis, it's not static. Yes. It's higher when babies are born and then goes down and perhaps it's slightly up. Perhaps you might be able to answer my question. But you don't think it's a physiological change relative to that? Yeah.
Yeah, I think that certainly could be part of it, the amount of astrocytes that are in the brain at different developmental ages.
Yeah. Thank you. So that could be physiological.
So a couple of things that, we just were talking about also stressing. So we have 40 patients that have undergone, about 40, it might be 42, 43 at this point, but around 40 some patients that have undergone these lumbar punctures. Not every family wants to put their child through a lumbar puncture, which is completely understandable. And I do have to thank Ionis Pharmaceuticals, who has really funded all of the spinal taps in this study. So just as a background, Elise's Corner gave us the initial funding to start the study, and we have the PT, OT, the physical therapy, occupational therapy, speech, swallow. You'll see that in a few minutes. All of that was funded initially by Elise's Corner, and then Ionis Pharmaceuticals has been paying for the procedures for this study for 7-8 years now. So all of these have been funded by Ionis. I will say, a lot of our adult patients have not wanted to go through a spinal tap. A lot of the pediatric patients, the families were way more willing to put their child through a spinal tap. So we don't have, you can see, we don't have a lot of adult data. So that is no pressure. No pressure. Just the facts that a lot of adults have not wanted to volunteer for spinal taps. That's okay.
In my experience, similar to other rare disease communities where you have a pediatric form of the disease and an adult or late onset.
Yes.
I don't think that I don't think that community experience is specific to Alexander disease. So
Right.
I want to make sure we call that out.
Yeah. We're just making the comment that that's really across the board in a lot of, research and diseases and things. So we had about 40 some patients undergo this, 21 of whom had elevated pressure at least some point during their journey. So many of our patients did have multiple spinal taps, and you can see that they're connected here. And so, for example, there are some patients in the middle, and you can mirror so people online can see, this patient had elevated pressure at the first, but lower on the second. You can see some of them go up and down, right? It's not static, and it changes over time. And then there's some people who start lower and then on a repeat are high. So it goes up and down. And that's another thing that I don't like to say we don't know or we don't understand, because it sounds like we're not smart enough to figure things out. But the reality is that there are changes that come and we can't really explain why over time there's a change in the increased pressure.
How GFAP gets into the CSF to begin with is something we've been thinking about for quite some time. And we don't have a good answer for it, and we're still working on it. And I think Ab got caught up with some of the conversations with Ionis, because it's a big question. And how does it go? Why is it going up? Why is it going down? And if you're using it as a biomarker, it really complicates things. So we'd like to know, and we can speculate to death, but we don't know. So
can I ask, is this kind of changes you're not expecting normal population? Healthy population, you don't see the changes, fluctuations. No.
Well, I mean, it's a little bit hard, right? Because we don't do spinal taps in kids that don't have problems,
exactly.
right? But I think many of us are of the assumption that your opening pressure is going to be less than 28, 90, let me say it this way, 90% of children's opening pressure is less than 28, probably less than 25. So there are 10% of the population that do have opening pressures that might be 35 or 33 that are a little above this, marker of 28 that are asymptomatic. And we know that because my colleagues at CHOP published a letter in the New England Journal of Medicine years ago about our experience. Those patients were being tapped for other reasons. Right, so they weren't just healthy volunteers. Healthy volunteers don't get a spinal tap, but so I will say the fluctuations in opening pressure do seem quite high, even when you're controlling for anesthesia and for other factors and position and other things. So we just are noticing that people talk about the paroxysmal regression in Alexander disease, where you can lose skills and regain them. I don't think that there's a correlation with opening pressure, but I do think it's the same idea where your opening pressure might be high, the next time it might be low, and then for reasons we don't know, it might be high again.
So in other diseases, this is just starting to be looked at. And so generally, a lot of pediatric natural history studies, they don't do LPs for other disease areas or they don't measure opening pressure if they do LPs. And so I think that this is kind of like the tip of the iceberg in terms of understanding in what diseases is pressure elevated. And I was reviewing the literature last year to look at some of this. And one of the things that was very interesting to me is that in spinal muscular atrophy, after nusinersen became commercially available, there are some groups that started measuring opening pressure in children with spinal muscular atrophy before they started them on nusinersen therapy, which is Spinraza, and found that there was elevated intracranial pressure in these children before they ever started the therapy. And that had never been looked at before. And so, it's just something that I think we're at the very beginning of learning about pressure in neurologic diseases of childhood, where there may not be symptoms, like Amy said, to clue you in that you need to look at the pressure.
I want to just I don't want to put words in your mouth, but I think for this group, why does this matter jumps to my mind. So I hear you, maybe one of the reasons that this research matters is do you think that this could potentially inform during the diagnostic journey in help narrowing down a differential, in addition to other ways like measuring drugs in trials, or why is this important?
So I think that one of the reasons it matters for me in terms of clinical symptoms, right? So I was not expecting the one patient that I mentioned who was rubbing the back of her head. Again, this is stuff that I learned from you guys, right? I never would have thought to ask if she was having headaches or ask these things. And as soon as we started diving into this a little more and she was rubbing the back of her head, I realized that I think she was having headaches and she just couldn't tell us, right? So in the younger children who are not able to articulate their symptoms, this is something that now I ask, do you think your child could be having headaches? And that's such an overwhelming question for someone whose child can't speak to them. But then when I say, do you see anything like them rubbing their head constantly or them rubbing their eyes, right? So I think it's important now because it made me realize that I need to ask the question in a different way, right? The other thing is vomiting, right? So we sometimes think about vomiting, intractable vomiting, being a symptom of increased pressure. And in that in the younger patient, we said, well, maybe that's the problem here. Maybe if we relieve the pressure, the vomiting will get better, and it didn't. So vomiting is becoming more and more concerning and more refractory to treatment in a lot of our patients. And just as a side note, the vomiting in Alexander disease is because in Alexander disease, there's a predilection for the brain stem, and there's a vomiting center in the brain stem. And so when that area is involved in Alexander disease, you get a lot of vomiting and this intractable vomiting. So there's a biologic rationale for the vomiting. But we were wondering, is it a pressure phenomenon? And so this was also important to me because I wanted to be able to understand everyone who comes to me with vomiting, not that I'm looking at anybody in particular. We're we're working on this nausea, and apparently mice and rats don't vomit, and that's what I learned from Tracy or whatever, but they have other behaviors. But anyway, so we're trying to figure out more about how to better treat the vomiting. And so one important question was, is that vomiting coming from increased pressure? And I don't think it is. I do think that, so to answer that, I really think that the vomiting is coming more from the area of the brain that's affected. But people who have come to me in clinic might see a line in my note that says, if vomiting is refractory, considering an LP to look for increased pressure. Yeah, because the management would be so different if that's what was causing it.
Yeah, just one question. Sorry. So this, as someone who's been coming to these for what I feel like is a decade, this is a new exploration in information. One of the things that comes to my mind based on personal experience is Olive has had, or potentially a patient has had 10 or 15 lumbar punctures. Where is that data? When will that data become available? Or is that data somehow being kept to be able to be released at some point in time to be used? It feels like Olive has as many dots on that map. She should already. And I think there's many of those. No, I know, but only the ones at CHOP. And I get that. But I think that within the community, there have been hundreds of lumbar punctures. Just a question. It might be an Ionis question. I don't know.
Yeah, I mean, I'll tell you. Sorry. So in our clinical trial, Ionis is an ongoing phase 1/3 trial of an experimental medicine called zilganersen. And in that trial, that drug is delivered through a lumbar puncture, and we collect CSF during that process, and we do other measurements during that process and throughout the trial. The data from that trial is being collected, and it's going to take quite a while for us to sufficiently and confidently go through it to make any claims or findings. So it's hard to give you a specific if that data, if you're talking about somebody who was in that trial, and it's hard to be specific about when specific data from that trial will be able to be released, but we have Yeah, I mean, Ionis has a long history. I mean, nusinersen we have a long history of publishing a lot of data from our trials. Of course, if anything from a trial is found to be related to safety and that drug goes on to be approved by regulators for commercialization, oftentimes regulators require any safety findings from the trial that are pertinent to decision-making by a healthcare provider to be included in the label. So I know that's not a direct answer, but yeah.
Well, let me say one more thing.
There's potential.
Yeah, yes. I mentioned that we published this just came out this month, right? It was accepted a month or two ago. So I will say that we were not measuring opening pressure in the clinical trial throughout, right? So we will not be able to go back to the beginning and talk about every single person's pressure over time that's in the trial. We will have some of that data because we've now added it. But we will not be able to go back and do a graph from the inception of the trial through present.
I'm sorry. Oh, no. My whole point is more dots is better.
Yeah, more data.
You have 10, but you can only use 5
well,
of the 10.
So here,
you have 5 of the 10.
Yeah, here's the other challenge. Our institutional review board. So this is all data, just so we're on the same. This is not data from the trial. This is from the natural history study. And I might have glossed over it. I don't remember if it's on this slide, but no, I didn't put it on the slide. We were only allowed to do 2 lumbar punctures as part of the natural history study. The IRB, the Institutional Review Board, who protects safety, said that we would not be able to do more than 2 because there was no benefit to the patient. Even though there's benefit to the community, there was no benefit to the patient. So these are 2 that are roughly done 12 months apart or longer. There's a couple of times where you see more than 2 dots. Those were patients that had a lumbar puncture for clinical purposes. We were allowed to use that data, but we were not allowed to do research only more than 2. So we right now, we can't do a study where we look at like 5 or 10 lumbar punctures in the natural history. Now, this is where AxD can come in. You know, if AxD writes a letter to the Institutional Review Board and says, this is important for our community to know, we need more than 2 data points here, then they would consider it, but that's still a several-year study. And as I will tell you in a few minutes, our funding for these lumbar punctures through research is coming to an end. So we'll go through that in a second, but.
Can I quickly ask? When you tested these 2 points, is at the same time of the day every person? Because the reason I'm saying is when my son was born, he was born with a severe hydrocephalus, and he had a fontanelle open. So we had a home ultrasound, and we were taking pictures morning and day and night. And the ventricle size changed significantly to the point like slit to it really expanded. So I felt like there's natural, exactly.
I will say it's very hard to do anesthesia and everything else and to time everything always at the same time. So no, these weren't necessarily the same time of day, but in general, we had a standing MRI LP slot at 1 p.m. in the afternoon. So many of them are going to be between 12:30 and 2 p.m. because that's when we had negotiated that spot.
Do we know if outside influences affect the CSF pressure? And are you guys measuring the atmospheric conditions when you do these pressures?
I have no idea. It's a very interesting question, but you can, all of us can temporarily increase our intracranial pressure if we sneeze or cough or squat, bear down, and it's dynamic. It is not static. You know, and so it's constantly changing. I think there's some really fascinating literature out of Germany, which looks at the respiratory cycle and how the respiratory cycle impacts the flow and your pressure and all of those kind of things. And so it is not something where your level is going to be the same moment to moment, day to day. We generally, when these are done clinically, you try to do them in the same way with, you know, not putting a lot of pressure on the abdomen, that kind of thing. But, you know,
should we bring this study up when we do the FDA meeting?
I think to be determined. I do think it's important, and I will let you guys decide what you want to talk to the FDA about, but it might be important, when people see increased opening pressure, they do get worried. So it may be important, and maybe Rob can give guidance too, to say that this is part of the natural history study, right? 21 out of 40 untreated, right? This is not the clinical trial. 21 out of 40 patients had elevated opening pressure at some point and without clinical symptoms. And so it's something that we now know is part of the natural history of the disease. So if in the trial or stuff there's data that suggests increased opening pressure, we have to think very carefully about, is it related to the disease? Is it related to the drug? Or both. All right, I don't want to continue to monopolize things. Okay, I'm going to go very quickly through additional publications so you know everything that we're working on, and I have to give credit to some of my colleagues in the room. So we these are all published as well. There's a brainstem chipmunk sign, which is a diagnostic imaging clue that is present in all of the subtypes of Alexander disease. We can talk about that. Dr. Messing had led the charge on this genotype-phenotype association for 14 common variants in Alexander disease. That's also published, and I believe he's already done a podcast on that paper. We worked with Ionis Pharmaceuticals and Dr. David Lynch from the UK on Are You Missing Alexander Disease? So the diagnosis in adults. We also have 2 additional studies coming out of CHOP, clinically important endpoints in children with leukodystrophy, a multi-site study. That is part of our GLIA CTN, our bigger network, and that is looking at time to event. So at what age do people develop symptoms? And then the identification of novel biomarkers of disease severity in a Cardi-Gutierrez syndrome and a different leukodystrophy, but Alexander disease is considered a control group, and so there's some GFAP and NFL data in that paper. So these are all published. These are the ones that we're submitting this summer. Did anybody have questions on the? Okay, This summer we're publishing on a dysphagia instrument to quantify swallowing. We have a paper coming out about language skills, expressive and receptive language. I have to give Francesco a shout out for doing all of the work to validate our gross motor function paper coming out hopefully later this summer in Alexander disease, and I hope he gets a chance to talk about that. We looked at the PDCAT, the Pediatric Evaluation of Disability Inventory. I'm excited about this paper because I really think that it supports what our colleagues in Italy are going to talk about next. I think looking at the different trajectories of disease, that wasn't the purpose of the paper, but I think it really lends itself nicely to complement your work. And then relation of clinical phenotype to GFAP concentration in CSF and blood in Alexander. So this is the GFAP paper that we're really excited about. And then we have a standard of care guidelines that everyone has been really excited about. And we're going to be publishing that hopefully in neurodegenerative something cases.
So, and for the patients who are newly diagnosed, why that matters, right? So a lot of times we're diagnosed and we're sent away. I was sent away with a sheet of paper with 3 sentences. So what this is going to do is this going to allow us, you know, as an organization and as a community, right, to say, okay, here are some things that you guys need to consider, right? Here are some different specialists that you might need to kind of have on your team, you know, and that's really empowering, right? Because you don't know what you don't know until you're thrown into a situation. And just having that standard of care guideline is going to help us be advocates so much better, so much earlier for our loved ones. So I'm really excited. I think we got a sneak peek last summer and we're excited about it. So, and we as AxD will make sure that we're kind of communicating that outwards to everyone. So.
What does GFAP stand for?
So as you saw me texting, my husband and son just arrived. So I'm telling them to come to the lilac room so they can hear me talk. So they get tired of hearing me talk. All right, GFAP is glial fibrillary acidic protein, right? And what we can, Tracy, do you want to answer? I don't want to monopolize. Do you want to explain GFAP?
Well, GFAP, just like we all have skeletons on a microscopic level, your cells have skeletons. And GFAP is part of the the skeleton of your cells. It helps form the structure. And so if you change some of the components of that particular protein, it kind of screws it up in a way where it no longer forms a network of filaments that support the cell, but it folds on itself as a ball and an aggregate. And so that kind of disrupts its own function, but also it aggregates in such a way that it affects the function of other components of the cell. So the cells that it's expressed in are called astrocytes. And so we've all been talking about that. And when the astrocytes get upset because they have this protein globbing up inside of them, they become reactive and they start becoming inflammatory. And so they might affect other cell types in the central nervous system in an adverse way. It's meant to be an injury response, but if it happens for a prolonged period of time, the astrocyte causes problems. And so we think that's what's happening with Alexander disease. Does that make sense? I'm hopefully.
As an Alexander patient myself, do I have GFAP? Okay. In a way, yes.
So it's a great question. So this protein, it's not the protein you eat, but this protein, GFAP, is in the cells in your brain, right? But we can't measure that, right? So we followed Dr. Hageman's example, and we tried to measure it in the fluid around the brain and in your blood. Now, the interesting thing for this paper, number 5, is personally, I think that the GFAP levels in the spinal fluid, I think they're a little bit more important, but blood is easier to measure, right? And we don't, and to Dr. Hageman's point, we don't know how it goes from the brain cells to the fluid to the blood. right? And then what does it mean? If it's higher in the blood, or if it changes in the blood, what does that mean? So these are all the questions we're trying to answer, but these are also the questions that you all, giving your samples, giving your blood and your spinal fluid, have really helped us try to understand. And it started, so Dr. Hageman, your first papers came out about that in 2013, 2015 in humans? 2015 maybe. Yeah. Yeah. So 2013 or 2015 is when some of these early papers, and now they measured it using in human, it probably 2015. So it was in a specific assay. We're doing it in different assays, but Olive, it's a great question. All right, just to wrap up, we have more papers coming up. Phenotypic variability in Alexander disease, because patients are so different, we need to make sure that the community understands the differences. Mosaicism, so I have to give Carlos a shout out here. We're trying to understand a little bit more about the genetics and how patients are affected. Imaging, cognition, some more imaging papers on these masses in the brainstem, and GDF-15 levels following Dr. Hageman and her lead in terms of looking at not only GFAP, but another biomarker called GDF-15 and trying to understand that. So a lot of different things that we're working on, all from the natural history study, and all grateful for the funding that has led us here. Additional projects, which I won't go all into, looking at seizures and vomiting and sleep apnea. So lots of additional things that we're working on. And just to try to quickly wrap up. So I'm going to skip over this just in the interest of time. Well, maybe not this part. I guess I should go over all of this. So as everyone knows, Ionis has funded this. Funded began with Elisa's Corner, took over by Ionis. Grayson's Ladder had participated in some statistical support and some private families. So this has been funded by many different patients and many different groups over the last decade. But a lot of our funding is coming to an end. So we're trying to figure out how to continue the study, how to focus the study to get really what we need, but reduce the cost, reduce the time that families come to CHOP, reduce the in-person visits. So just to go over one other thing, our NIH study. So the Myelin Disorders Biorepository Project, the GLIA-CTN, that 5 years turned into 6 years of funding has expired or is expiring as of Monday. There is a renewal that the NIH is reviewing that we're hoping will be funded, but with the NIH right now, things are in flux. But we are able, and Francesco can talk later about it too, we are able to do some of the testing also under this study funded by the NIH, which makes us happy that our tax dollars are going to fund Alexander disease. Okay, so this is what's changing. Clinical visits are the same. People can come see us at CHOP for clinical visits. There are no differences, and those are billed to insurance. That's no different. Our commitment to Alexander disease research is not changing. We will still do Alexander disease research with the ability to do all these tests or not. The only thing that is changing is that we're going to reduce the number of tests that people have to come to CHOP to do. We might add in more remote visits to see if we can do things remotely so that you don't have to travel. We're trying to collect more data through questionnaires and surveys rather than having people come and meet us in person. And the only major change that will affect families is that the travel reimbursement will go away. So right now we've been able to, thank you to Ionis, that we've been able to fund travel to CHOP to participate in the study, but we won't be able to have that travel support after December 31st. So people still can come. We can still do a lot of the research at CHOP. They just won't get the travel support, which we know many families are dependent upon. Anything that families should be doing, well, if you want to come to CHOP in the next 6 months, let us know, because we need to know right now and get in before December 31st. There's a lot of ways to participate. Here's our general team email. And we are still applying for new grants and new funding sources to try to be able to continue the funding indefinitely to further Alexander disease research. Okay, the last thing is I feel like we've lost some of the community engagement, right? I feel like that research has become a burden. People don't always answer our texts or our emails and whatever. And so we want to be able to rally the community for support. We want to come together. We want to make sure, and that's why I think these meetings are so helpful, because I do think we leave with a renewed purpose, and then we want to then, get more involved and get more people involved and stuff. So let's change this narrative Let's involve our adults more. We don't take advantage. We're always so focused on the kids, as we should, but we're not involving our adults. And you might have been on our list to come in for a research visit. We just hadn't bothered you yet. But come in, see us. Let us collect more data. Let us collect more information on adults so that we're not just pediatric focused. And I think I'm just going to stop there, other than to thank our team, thank my son and my husband for putting up with us, and thanks to all of you. Anybody have any questions for me?
No, but just letting you know, I'll be emailing you before
Great.
December, obviously.
We look forward to seeing you.
Thank you.
And please too, I encourage, reach out to AxD. We're the global support organization for this disease. We can help point you in the right direction. We can make sure that you have information that's correct and relevant for what you're going through. So I'm not sure if you're on social media. You can follow us on, I think, Instagram, Facebook, and AxD. And our our email too is info@endaxd.com. Org. So info@endaxd.org. And we can help put you in contact with Dr. Waldman and just a whole host of other things too.
Perfect, thank you. And a trial still undergoing? Like, are you taking anyone else on?
Enrollment is closed.
Sorry, I want to give her a more complete answer. Yeah, so this is Rob. To the person who asked the question online, the Ionis phase 1 to 3 trial of zilganersen is currently not enrolling additional patients at this time. It will not enroll additional patients. However, there hopefully will be additional clinical trials on additional investigational products in the future. So the best thing to do is to continue to express that desire to your doctor, because who knows when the next trial will open. So if you're interested in it today, make sure your doctor knows you're interested in it, so that if there is a new research opportunity, they get to you right away.
Okay, thank you.
Is the research study, like you can only do one or the other, you can't do both?
Correct. Patients that are in the clinical trial are not eligible for the research study at CHOP. We can't do blood work. We can't do spinal taps on those patients. We can't collect surveys, right? So those families don't get a lot of communication from us, because we don't ask you those things because you're in a different category. So those patients can't participate in the natural history, but other patients still can.
The meeting that you guys had at CHOP last year, I thought was, from someone who doesn't have the experience that a patient does, I thought it was terrific to hear all the resources that were available. And I know there was some talk about, and I don't want to put anybody on the spot, but maybe this is what you were getting at. There was some talk about trying to put together a package of information for folks with newly diagnosed patients in terms of resources. And I just thought, if that's coming out, that'd be terrific.
So I was actually just approached a week ago by a journal who is looking for wanted us to write a paper on the future of Alexander disease. And I said, I don't have the bandwidth to write that right now, but the journal's called Neurodegenerative Clinical Management. And I said, I have a standards of care paper. Are you interested in that? And they said yes. So we're hoping to get that paper published by the end of the summer. And that paper would then accompany this document for families in terms of what we envision is we would post that paper on the website. Now, we were going to submit it. It sometimes takes a couple of months after you submit a paper for it to be accepted, but hopefully by the end of the year. So that paper would be available, and then we would post it on EndAxD with additional resources like physical therapy and occupational therapy and that patient guideline.
Yeah, absolutely. And also what we did, so I don't know if anyone's ever sat down, with some free time and dissected Dr. Messing's book. It's a lot. And quite honestly, to a family who doesn't have a medical background and who's being faced with this diagnosis, quite honestly, it's too much. So what we did, and what Ionis helped provide us with some funding for, is we took that book, and I think the average American reads on a 7th grade level. And what we did was we broke it down so it could be more digestible, just so patients could get the information that's really important, but kind of take it down to the right level. So I think the standard of care combined with this, it's called Understanding and Empowering AxD. It's pictorial too. And then ultimately, as we continue to evolve, starting to put together, lists of, physical therapists in your area, OT, speech, all that kind of good stuff. So this is just the start. And I think that we've made some pretty good progress though. Maybe we did. No. So, and people on the online can't see it, but it's called Empowering and Understanding: A Comprehensive Visual Guide for Alexander Disease. And if you notice, there is the rare zebra butterfly. So the zebra is the yeah, but we like the butterfly for a sign of hope. So it's all throughout there.
All right, I'm going to pass the mic and the slides. Oh, you're going to want to keep the mic. Here.
Hi everyone again. So these are my disclosures. Ionis is helping us as well with some of our projects. And so I will start with some words about Alexander disease itself. I'm glad to see there's some new families, so maybe that will be helpful for some of you. So let's just imagine our brain like a city. So that's full of roads that leads you that connect places. So the roads may be like the neurons, like the essence of our neurons, and these roads need to be kept safe from some barriers, and that may be the myelin. But we don't have just myelin and neurons in our brain. We do have many other cells that help, like keeping the road clean, keeping connections, bringing nutrients to the other cells. And some of these functions maybe are exerted by some cells that are called astrocytes, as Dr. Hageman was trying to explain before. And these are these star-shaped cells that that are mainly affected in this disease. How are they mainly affected? We were talking about GFAP, that's the gene that's mutated in Alexander disease, and these pathogenic variants in GFAP cause an accumulation of this protein that's misfolds and accumulates in astrocytes. And not only does it create damage, like impairs astrocytes to do their job, but it also creates some mess around, some neuroinflammation, and some degeneration of the myelin itself. So how does it affect patients? So clinical manifestations are very broad in this disease, starting from the age of onset. They may be from neonatal stages of life to adulthood, and now we know also older adults. And symptoms may vary from like enlargement of the head, that's macrocephaly, to developmental delays, seizures, but also abnormal eye movements, difficulties in swallowing, difficulties in talking, nasal speech, difficulties in controlling palatal muscles. And this mess that GFAP is causing can also be observed at the MRI. What is MRI? MRI scans are those pictures that we are doing of our brain. And just to give you an idea, so we have like white matter in the brain and gray matter. So white matter would be the thing that's inside, and gray matter is the outline. So in an healthy brain, in this kind of images, MRI images, white matter should look like this, so black or dark gray, while gray matter should look clearer. So what you see here in Alexander disease is that it looks pretty bright. that's what we call signal abnormalities. That's what we call signs of demyelination, and that's what happened in Alexander disease, where the pattern is actually typical of involvement of the anterior, that's frontal, the anterior white matter, more than the posterior. That's you see pretty black here. Additional findings are also typical. Dr. Waldman was speaking before about the chipmunk sign. That's the involvement of brainstem, but also basal ganglia that are deep structures in the brain are also pretty much always affected. And contrast enhancement is also often present. Contrast enhancement is kind of a sign of brain inflammation in some ways. So that's a very complex disease, and also we are really going forward with research. There are many investigational products under that are studied in several labs around the world, and that's currently an investigational product under clinical trial investigation today. So the main problem in this era of incredible innovation is how are we going to interpret the data in this clinical trial setting? Do we know enough about the disease? Do we know enough about the disease course? Do we have enough biomarkers to understand whether a treatment is effective or not? And so we started kind of a long journey in this disease, starting from the phenotype. So what does phenotype mean? Phenotype is the set of observable traits of an organism. And how do we phenotype Alexander disease? We need real-world data. That's real-world data about the clinical presentation, disease trajectory, developmental milestones, clinical symptoms. And this is what will give us an idea of the phenotypes in Alexander disease. And so starting from the phenotypes, starting from the patients' stories, patients' clinical trajectories, we observed that some of our patients shared some disease trajectories. We also just took into account type 1 patients, so because we are pediatric neurologists, and that was the only cohorts that we got to know in clinic. And then we try to apply the findings that we came to from our cohort to a cohort of patients described in literature, of which we had enough clinical data. What we came to was that there are kind of two main groups of patients. Like we could divide, so four groups in total, but we could divide them into two groups that show a more severe clinical picture with no acquisition of autonomous ambulation, and two groups that show a less severe clinical picture characterized by the acquisition of autonomous ambulation. And among these two pairs, we have both more slowly progressive courses and more rapidly progressive courses. So we would have a type 1A that doesn't acquire autonomous ambulation and really rapidly deteriorates, a type 1B that doesn't acquire autonomous ambulation but has a much slower clinical course, while among type 1C and type 1D, we do have some patients that acquire autonomous ambulation but then more rapidly decline, and some others that pretty much remain stable across adolescence. So we were wondering, why does this happen? So we know that one gene is responsible for the disease, and we also know that very often patients with the same mutation have very different clinical pictures. So we tried to analyze the genetic data of some of our patients, actually patients from a broader cohort of international patients, and some others derived from literature. And we kind of arrived, like we kind of did similar work to what our colleagues Dr. Messing and Waldman did of reviewing on genotype-phenotype correlation, it was a much broader, theirs was a much broader study that included also others. We just focused on pediatric patients and on their clinical evolution. But we kind of came to the to very similar conclusion actually, because we observed that like there are a consistent number of variants that are private, what we call private. That means that just one patient has that variant. But there are some variants that are more common across subtypes. And the most common in our cohorts were these ones. So R would be the letter to define an amino acid, that's arginine. And so arginine 79 and arginine 239 were the most common affected. And the most common changes were cysteine and histidine and cysteine and histidine. And what we observed is that R79 was most commonly observed in less severe affected patients, while R239 was mostly present in in more severe phenotypes. The changes also helped us in a way. So the histidine changes, like they acted kind of differently in the two groups. Like the cysteine was much more present in patients that were also severe in R79, but in the changes in 239 was not really so informative, because we could have that across a lot of subtypes. So what was very striking was that the change in histidine at this level was very much more present in very severe clinical pictures. So, and what we also observed was that actually like our disease trajectories really like correlate well with the age of onset, that was increasingly high with the less severe phenotypes. But genetics tells us something, as you've seen, but not a lot actually. So we just saw 2 variants and yeah, some kind of correlation, but that's not much actually. So what we tried to analyze, what we tried to understand was also actually whether there were some neuroimaging features that could help us kind of predict what would be the clinical evolution over time. And we started from the observation that actually in our patients, the MRIs kind of looked really different from one patient to another. So what we did was create an evaluation protocol that evaluated a lot of areas in terms of areas that are affected and presence of mast lesions that were mentioned before by Dr. Waldman. And so we kind of found that there were some areas that would suggest the possibility to develop a much severe phenotype compared to others. Just to give you an example, I put up some MRIs from a 1B patient and a type 1D patient. Like, for example, here, you can see, like, this is the temporal lobe. You can see that's all white, for example, and here it's all black. So this was an area that was shown to be statistically significantly associated to much severe phenotypes. And also, for example, like more posteriorly here, you can see in much less severe patients that's all black, and here that's all white, and that's another area that was significant in our study. What we also observed was that also changes in what is called cerebellum, that's another structure of our central nervous system, was statistically significant, and it was like, here you can see the white, here there's no white, and also at the level of the corpus callosum. So these are additional findings, as I was mentioning. So the brainstem was pretty much observed, the involvement of the basal ganglia was pretty much observed in all our patients, and the brainstem as well. And here you can see a lot of nice chipmunks.
So what we did with this, so these were the 6 areas that were shown to be significant. What we tried to do with this was kind of create a score that could help us from the analysis of the first MRI to kind of direct us towards a more likely disease phenotype. So we assigned a point of 1 for when the area was affected, a point of 0 when the area was not affected, for a total of 6 out of 6 when all the areas were involved, and 0 out of 6 where no area was involved. We observed that for scores higher than 4, there was a significantly higher risk of being a more severe phenotype, while for scores below 2, it was much more likely to have a less severe phenotype. So we kind of tried to create a flowchart combining these MRI findings with what we think it's growing important, that's the acquisition of developmental milestones. And what we came to the conclusion to is that one could analyze the MRI, and with the MRI scores above 4 should look at acquisition of head control. Acquisition of head control, so some posterior acquisition, would point toward a less rapidly evolving clinical phenotype, while no acquisition of head control would point towards a more severely evolving phenotype. For scores below 2, what we suggested to analyze was 2 additional areas that were shown to be significantly differently associated to a more rapidly evolving, less severe phenotype, so acquisition with the acquisition of autonomous ambulation. So involvement of these 2 areas would lead to a type 1C, while no acquisition of these 2 areas would lead to a type 1B. For intermediate MRI scores, we would look at autonomous ambulation. No acquisition would point towards the more severe end of the spectrum. Acquisition would lead to the less severe end of the spectrum, and then the other 2 things that we have already mentioned should be again evaluated. So this is about MRIs. We are looking for other things that may modify the phenotype, or maybe something that could be used for additional standards of care, disease management. And what we're also going to analyze now will be the gut microbiota. What is gut microbiota? We have our gastrointestinal tract is colonized by a huge number of microbes that changes across our lifespan. So it's influenced by a lot of things like maternal factors, maternal diet, birth method, feeding method, smoking exposures, use of antibiotic, geographic area of residency, and physical activity, for example. And what is becoming more and more known in recent years is that there's a tight communication between our gut microbiota and our brain. In particular, we know that gut dysbiosis that may be caused by gastrointestinal symptoms, such as the ones that are very frequent in neurological disorders, but in particular in Alexander disease, could cause the production of some metabolites, some substances that influence in some ways the brain, and in particular they contribute to brain inflammation, and at the same time brain inflammation influences gastrointestinal symptoms and gastrointestinal health. That's what's called a bidirectional communication, or also gut-brain axis, and it consists in a bottom-up communication and in a top-down communication. Why is this so important? Why is this relevant to Alexander disease? So very recent study has shown performed in mice, in particular in germ-free mice that are mice without microbiota, is that they have some they may have some altered myelin production in a certain area of the brain, and also it has been shown that also treating healthy mice, so mice with a healthy microbiota with antibiotic, would affect myelination process. So given these connections, what we are going to do is analyze feces sample of some of our Alexander disease patients to understand whether they there is a difference in expression of some genera, some type of bacteria that is relevant and is different from control samples, and of course whether this is related in some ways to the patient's clinical evolution over time. So going just, I also wanted to just highlight how important is patients' involvement and patients' experience in our research, because we always try to kind of set research priorities based on what really matters to our patients, and of course also like sharing. Thank you. And also of course sharing their data. Of course, it's very important to us because it's an ultra-rare disease, so numbers matter. So the more people take part in research, the more reliable the results are, and of course also biological samples. And that's just kind of a diagram of how things, how research works and how things go forward with research. Like, it all started with a question that a doctor cannot answer, and that doctor then starts to ask questions as well. So as patient data, samples, and then analyzes those, and maybe is able to get back with an answer, and then it all like moves in circle and helps research proceed and move forward. That was actually all of it. I'm happy to take questions, and this is Filippo. He would have loved to be here today, but his mother didn't want him to take this long journey, but yeah. And this is the Italian Association, Alexander Disease Association. That's an incredible patient advocacy group, and it has been a support for a lot of newly diagnosed family. And thank to you all, and thank to our team in Italy. And
so you say, you know, the importance of getting involved in research, right? So, I mean, how do we here, I mean, how do we help support you guys?
Yeah,
what does that look like?
Yeah, so of course, like we love to kind of have our first kind of conversation with you every time we get to know you, like just to know about your story, your clinical evolution, how the disease arrived to you, and how it progressed over time. Because like the first thing in my opinion in this disease in particular is the clinical evolution. That's very heterogeneous. And also the samples. I'm working on it actually. Like Thomas is I heard from him quite regularly. I'm trying to get our IRB be able to receive samples from abroad, because that's not something that we can do yet. So yeah, our Italian patients are very cooperative, and we got to have all of their samples. They got to do skin samples, so they're very cooperative. But yeah, we are working on being able to get samples from abroad as well. Yeah.
So thanks for the talk. That was actually really good for the non-scientist here. Why is Italy studying this? There are a lot of cases of Alexander's disease in Italy. And the second part to the question, are you guys pushing to bring this upstream to the EU and get EU funding research dollars?
So to us it's a bit easier kind of for like assessments and everything, because also research assessments are funded by the public system. So we're not really we are not really so familiar with regulatory things of funding in this yes at that level. But as far as the start of the question was if we follow a lot of patients. I mean, that's not specific to Italy. We've been getting patients from all over Europe actually currently because of the trial. They got to know our leukodystrophy center, and actually our community, like the patients we follow, has grown consistently in the past 2 years. But I don't think that's something that's related to Italy. It's just like that the disease is becoming more easily diagnosed even in centers that are not specialized in leukodystrophies. So when they are diagnosed, often they are sent to us because now we have whole exome sequencing. Sometimes people just do whole exome sequencing for some developmental delays, and then they end up with these variants in GFAP, and then their doctors just come to us and say, oh, you know something about GFAP, please can you like follow the family. So yeah. Thank you.
It's weird. My question that I'm posing to everybody, including those online, to you, is how do we, in search of additional families, additional caregivers, additional patients to participate in our presentation of our community to the FDA, but also to each other, how would you recommend we best connect with your patients that you know that are not here and probably are not listening? Is there one of these there that you'd like me to come to?
Yeah, that would be lovely. Yeah, they're fantastic people, and I'm sure they're willing to kind of connect. I think an obstacle could be the language, actually, and that's why a lot of our Italian patients do not end up like connecting with you because they don't know the language. But of course, I think it would be great just to kind of be in touch and like share projects, share experiences. We have sometimes also had patients who didn't want to connect, actually, because of course they know that every patient shares their story and that stories may be very different, and so they were like, I don't want to like know that one patient is doing this while my son is not doing that, and that's also okay. It's just like I mean, I think it's just important for all families to know that you are there, and that if they're willing to connect your
What is that, a Alexander disease specific organization? Is that leukodystrophy? What does it translate to?
Peter Pan.
More unique than rare.
Oh,
Really?
I should have guessed that.
What's the significance of is that Peter Pan?
Yeah.
Oh.
Do you know if the organization works with the European Leukodystrophy Association?
Not really. It's a small association, actually. They do a lot of work in Italy. They fund a lot of projects in Italy. Actually, they are Alexander disease specific, but they actually fund a lot of things like for pediatric hospitals in Italy, and also not Alexander disease related projects, but yeah, they're quite a small association. So I'm not sure they really know what, for example, ELA is, or yeah.
They're like Italy has a long history of having some really well organized patient advocacy groups in a lot of different diseases. A lot of people don't realize that, but they're one of the countries in Europe that just has done this really, really well. Happy to connect them to other groups that have learned how to collaborate internationally.
Yeah.
And they can figure that out.
And Jay, to your question about how do we connect with them. So we actually have a member of our community who lives in New York. Her name is Chiara. She has a little girl with Olympia. She's from Italy. And she's actually offered, and she's looking for ways, or she has been looking for ways in the past of like, what can I do to bridge the gap, you know, language-wise, to help connect to people kind of over there. So that, I mean, all of it sounds like a good opportunity that we can follow up on, because, yeah. As if we needed one more complexity with this disease, right?
I kind of wanted to ask the same kind of question. Can Japanese patient population, can we help your research somehow get connected? Although, of course, there's a language barrier. But, you know, as you said, there is microbiome that are different based on different diet. And, of course, we have different diet in Japan, and thought it would be interesting to compare.
Yeah, you're totally right. That's a great question about the diet. We in this project, we just asked for a patient that they didn't have any corticosteroids or antibiotics before 3 months before the feces sample acquisition, and we also like kind of take their nutritional status and their diet routine before like taking the sample. And that would be great actually, but as I was saying, we're working with our IRB to be able to get those samples from abroad because it's still not regulated. But another thing that could be very helpful would be the MRIs, because we are just trying to apply this to another cohort, because of course we have kind of tried to auto-validate the flowchart on our cohort, but it's kind of biased because we created the flowchart on our cohort. So that would be lovely to kind of like validate it in some other ways.
Let us know.
Yeah, I would encourage Alexander disease researchers considering this cross-country collaboration to pursue it, number one, but at the same time recognize that there are that there are many other disease areas that are way ahead of you and have solved these problems before, and I would encourage you to leverage their lessons. The Alzheimer's community has done this really well. MS community has done this really well. ALS, MND has done this really well. So as you run into these problems, they're all solvable, but be willing to kind of ask for help and not have the drive to innovate where innovation isn't necessary.
Can I ask if trials are still ongoing in Italy?
The trial is still ongoing, but it's not recruiting. It's enrollment is closed. I'm sorry.
Is that for everywhere? Does anyone know of trials anywhere at all?
Sorry, do you mean the therapeutic clinical trial, or?
Is the question about interventional treatment-based clinical trial or about research trials overall?
Yeah, clinical trials.
So if the question is about interventional treatment-based clinical trials, at this time, I don't think anybody in the room is aware of an actively enrolling clinical trial. Doesn't mean it doesn't exist. We don't know everything in this room, but currently there's not something that we're aware of that's enrolling. That, like I said earlier, that changes oftentimes without announcements. So I would just say continue to talk to your doctor, monitor the news, make sure you're on AxD's email list, because they'll blast that out when that happens.
Okay, thank you.
Yeah, and also another thing that we found it's important regarding this thing is like always be connected to a center that knows about these things, because some people may lose their opportunity like timely because their doctor was just not aware of it, and just stay connected to centers that know the disease in some ways.
And we're going to hand it over to Yukari for her presentation. For our online folks who's asking that question, obviously it can be very upsetting hearing that there isn't an active treatment trial, but that does not mean there's not active things that we can do to help and to keep your loved one healthy and thriving and all those other things. So please, talk to your medical team. There's a lot that you can do in outside of a clinical trial to be well. Turn it over to you.
All right, thank you. Can you start the slide? Yeah, and I'm aware some of you guys will probably have to catch slides. So yeah, no problem. Thank you so much. And I'll make it really short because I know that we are way beyond our timetable. Yeah, I did, yeah.
You had it up once before.
Well, there we go. So again, thank you for being here today with us. I'm Yukari, and I'll be talking about some of the updates on UMass, University of Massachusetts research overview of AAV-mediated gene therapy for Alexander disease. I had a lot more, here's my COI. There's a lot, but I'll be speaking from the perspective of an individual from the AxD organization. So I realized that we are really deep into this community, and we don't really have to talk about the first potentially 3. So I'll go straight into the last topic, which is basically the updates. That's Kuno and myself. So yeah, everybody here is very much aware of Alexander disease. I'll just skip over. And are we okay with what AAV-mediated gene therapy is about? Okay, sure, yeah. So it is gene therapy. We have a construct to help rescue the erroneous gene that we talked about, the gene that causes GFAP. But in itself, we can't really deliver to the place that we want it to be delivered to, which is astrocytes in our case. So we have to put that, basically the active drug, into something that can deliver to the brain, which in our case is AAV capsid. That's basically like a small cargo which contains that AAV genome active drug into where it should be. So this is a construct. And please interrupt me and with any questions. Yeah. So again, you design the genome, artificially designed genome, and package into the cargo, which is the AAV capsid, and dispense into fluid and inject in different ways. So we know there's an intravenous injection. We have ICV, which is direct injection into the brain. We have also other ways that's like a spinal cord injection and so on. So we hear about gene therapies a lot nowadays. It's becoming more common, but it's still fairly new modality compared to like small molecules, which is like a tablet form to take, and other modalities. So there's inherent uncertainties and pros and cons to it. It is a viral vector, even though it is not infectious. So there is safety concerns. It is not infected, but that doesn't mean it does not have any risks. For example, you might have heard about immune response when the AAV gene-mediated therapy is injected. And like I mentioned, the cargo has a destination where it should be delivered to, but it's not perfect. So sometimes it gets dislodged into other places within the system. For example, the top of the list would be the liver. And when there's a response, immune response, it causes troubles and sometimes causes liver injury. And it could happen to anywhere else, like heart or kidneys. So this is a safety concern. And it's a newer modalities and very costly and challenging technically to produce a quality product. So that's also one of the challenges. But we have a lot of pros. Well, good thing about this condition approach is that when you talk about pills or injections or even ASO, you need to keep on topping off because the efficacy of the drug doesn't last forever. But with gene therapy, this is not the case for everything, every condition, but the efficacy lasts for a long time. And right now, single dose should be able to last for a good few decades so that you don't have to redose gene therapy again. And the reason why this new modality is so expensive is because it takes a long time and a lot of money to develop. So from the top down, it shows the different steps for a new drug to be produced. And at first, there is something called basic research. Basically, you're coming up with a hypothesis, and you're understanding how this condition happens. So that you would have target candidates of 20,000 plus, sometimes even more. And that it takes about 2 to 3 years. And then you narrow down your hypothesis and the candidate list of the drugs or drug Cs. And this process is called drug discovery. And then you narrow down even further in the preclinical research, which means you're testing your hypothesis in living animals, like rats, mice, and so on. And that takes about 3 to 5 years. And you see the numbers getting smaller and smaller. And at the clinical research stage, it's now down to 10 from 20,000 plus. And that can take up to 7 years. And with the result of that, we apply to health authorities like FDA or EMA in Europe and PMDA in Japan. And that process itself could take up to 2 years. In total, we are looking at about $2.6 billion and 10 to 15 years for one drug to be approved and be commercialized. This is a really long process. And that's the reason why it's so expensive to get these drugs. Yes, please. Yes, so it's inclusive and on average. There's a wide range, of course, but yeah. Good question. Yep. So within the clinical trials, it means studies in humans. But there are different phases to it. And we start with phase 1, which means usually done in healthy volunteers. And unless it is a condition that's specific to women, it usually means healthy male adult volunteers. And the focus of this is safety and optimal dose, and PK/PD, which is pharmacokinetics and pharmacodynamics. So you want to see how the drugs is distributed inside your system, and you want to see how the drug behaves inside your body. And phase 2 is you're looking at the safety and tolerability and efficacy. You want to see if the patients can tolerate whatever the drug it might be, and you'll be testing in a very small group of patient population. And if everything went well in terms of efficacy and safety, then you move on to the P3 study, or sometimes this study is called pivotal study, because the data or the outcome of this study would be the data package that FDA, for example, would be looking at to see if they would approve this drug. But FDA approval is not the last thing. Then we can move on to the phase 4 study. Sometimes this is called a post-marketing surveillance. So approval is not the end of the story. We have to monitor actively if the drug is safe still to use and efficacious enough to use. So this is the same illustration. I just wanted to show the candidate drugs what we have for Alexander disease at the moment. The closest to approval is Ylenia's ASO or antisense oligonucleotide therapeutics. And then I'll be focusing on the UMass study today. And there are others that are behind this stage, but there are quite a few. Okay, I don't know if I just wanted to make a point that the reason why there's so much dilemma surrounding the gene therapy is because, like I mentioned, it's very costly to develop. And this modality is not for everybody. Sometimes people can have something called antibody to AAV itself, and those people will not have the benefit of this approach. They are not eligible to receive this therapeutics. And this is one time in life, well, at least now, for now, it's a one dosing per life kind of approach. So what does that mean for us to decide something this important, life-changing event for somebody who cannot make that decision for themselves sometimes? That's a lot of responsibilities. So then I'll switch the gear and we'll be sharing about the updates of UMass gene therapy on behalf of the research team, Jun and Guanping and B. So I mentioned about this specific cargo called adeno-associated virus, and in this case is a type 9, AV9, which transduced astrocytes efficiently, they found. They injected this capsid intravenously into animal models, and they successfully bypassed the blood-brain barrier, which is, you explained, Amy explained well, it's the protection for the brain, the central nervous system. So they crossed that barrier successfully, so they thought this would be an optimal way to deliver the active drug. And what you see here, the green part is actually the transduced cells in the brain. So you see it's dispersed pretty well. But again, this is an animal brain, not human brain. I'm not going to the details of the construct. So this is a schematic view of UMass AAV gene therapy approach. Again, they designed artificial mRNA to target the GFAP, that's our target, which is a causation of Alexander disease, and then packaged into the AV9, and then injected into mice or rats, Alexander disease model and healthy control through intravenous administration or direct injection into the brain. And they took a look at different safety, efficacy, and PK/PD measures, and I'll talk a bit about it in the later slide. So they looked at, for efficacy, the GFAP levels. We talked about earlier presentation that when it reaches a certain threshold, it starts causing troubles. So we looked at that, and we looked at weight changes and food consumptions. We looked at imaging such as MRI imaging and histology. Histology is basically you take a chunk of tissue from, or in this case is brain, and stain it and look at under special microscope. And then we looked at the functional improvements by rotor road and grip strength test and ambulation. This is just means walking. And for safety, we looked at survival over time, weight changes, and histopathology. Histopathology is something that's similar to the histology, except in this case, we're looking for signals that's concerning. And we looked at the heart, kidneys, colon, and liver. So they first did the mouse study. This is a busy slide, but basically I want you to just take a look at this part. You don't have to take a look at this part. That's just a reference point. And so the left-hand side, this is a healthy control. So this is not the disease model. And the middle is untreated groups, and the right is a treated mice. What you really want to see is the intensity that's close to, as close as a healthy control. And this is what we saw. And this graph quantified what we see here, numeric manners. So black one is a healthy control, and the red untreated, and green treated. And as you can see, the GFAP level for the treated mice are almost equivalent to that of the healthy control. So this is good. And this is how it looked like under the imaging. And this is a section of the brain with different parts of the brain, corresponding to, for example, this part, as you can see, is olfactory bulbs, which is an area that's responsible for sense of smell. As you can see, this is how the healthy control looks like, and you want to see as close as these levels of intensity. which you see in the treated model, but not the one in untreated model. And same for hippocampus. It's here and that. So they did successfully confirm the efficacy in mice model, but then they realized that it does not, mouse model really doesn't show the phenotype of Alexander disease that you would see in humans. So they moved on to the rat study, which is, again, not perfect model, but closer than the mouse model. So this shows GFAP expression in rats after 3 months post-injection. Again, this is a section of the brain with different parts corresponding to these. And I'm not going to the details of this. And all I want to tell you is that they saw overall improvements in the levels of GFAP, but not perfectly and not across the same sections. Some parts you see more effectiveness than others. So this is the challenge with the AAV gene therapy, because it doesn't really get distributed in the brain the same way that you would hope. And this is 13 months after the injection. Only thing I wanted to note was the trend is persisting, and 13 months in rats years, rats months, translates into a few decades in human years. So the efficacy is persisting, but not perfectly. But that was the case even at 3 months. And this is a graph for weight change, and the left side is, is a female, and right side is a male. And y-axis shows the weight of the rats, and x-axis shows the chronological age on top, the black ones, and the green numbers show the weeks elapsed after the injection. So you see the general trend. For female, for the first, I would say between the weeks of 4 and maybe 18 weeks or so after the injection, there's a slight recovery of weight. Oh, sorry, I should have said this black one is again the healthy control. The red one is untreated, and blue one is treated. So there's slight recovery. But towards the end, as the time goes, you don't see the difference between the treated and untreated rats in female. And for male, it's the opposite ways. At first, you didn't see the difference between the untreated and treated rats. But as the time goes, at the end, you see slight improvement or weight recovery in the male rats. But it's a bit disappointing to see not much recovery in that. Yeah, of course, yeah. Reduction. Not much, but that's a really good question, because that's one of the key areas. That's, yeah, of course, yeah, yeah. So yeah, there is some, but maybe not. Yeah. Oh no, no problem. Anytime. So that, and at first though, they just looked at the weight. So I asked if it was to do with the appetite, and so they started checking the food consumption, and they found they actually eat less, the disease model. So that might be because of that. So this is T2 flare MRI imaging, and I want to note that untreated rat brain has an abnormal signal, like here, which in the treated rats, you don't see it. So this is, yeah, effectively treating. Oh. So this is histology under the microscope stained. So it might be hard for you to see, but there are black arrows showing Rosenthal fibers. And so here's a healthy control and untreated and treated. Even though there are not arrows here, there's a lot more Rosenthal fibers you see here. They didn't put it because there are so many, and they just simply didn't. And these red stained parts are the Rosenthal fibers. So there is a bit less here, and the same trend in other parts of the brain. Again, this is other parts of the brain, corpus callosum and cortex and midbrain. Basically the same trend. These are the Rosenthal fibers, and you have much less here. Yeah. Same trend. And this is 3 months after injection. Same areas of the brain and much less after the treatment. I think you can agree with this. And this is olfactory bulb. Again, the same trend. So this is a functional test. And first they did rotor rod, which looks like this. Basically, you place the animals on the rotating rod, and you want to see how long they stay on this rod. So higher the number, the better performing they are. And so you see the black dots are the healthy control, and the red, again, the untreated, and blue treated. So you can see the treated group performed almost as good as the healthy control, the ones without disease, for female and male. And then there's a grip test, which basically you let the rats grab this apparatus and let them pull and check the strength of the grip. Again, black denotes healthy control. Is everything okay? Something funny? Really? Really? Okay. Well, it's pretty simple, I think, straightforward. Yeah, it makes sense, right? Grabbing the thing. Well, here it is. Yeah. Yeah. Did it work well? Yeah. It's straightforward. You just yeah. 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 It's their natural instinct, exactly. Yeah. They do not do it. But yeah. But again, as you can see, this is different time points. And this is a female on the left and the male on the right. But basically you see the same trend across sexes. The treated rats performed almost as good as the healthy rats. Oh, sorry. So, okay, it started already. So that's the healthy rat. So that's how it should be. And this is treated. And this is untreated rat. Yeah, I know. So I hope you could see the difference and get excited as I am. Yeah. 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 So that was tested at 8 weeks in the 3 weeks old rats. So about 5 weeks after the injection. Yeah. It's a juvenile. Yeah, so Amy asked the 3 weeks old rats translates to what age in humans. I know there is a range, I believe, and it's a juvenile in humans. Yeah. 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 Oh, yeah. Okay, I think she's right. Yes. Yeah. Process is much longer. Yeah. Oh yes. Yes. Yeah. Young, yeah. Yeah, thank you. So we'll look again. Wait, okay, this is no. Can you start the healthy and then untreated and treated? Do you think? Oh, sorry. Do you think you can start from your end? Oh, it's okay. So that's healthy, healthy rat. Yeah, there's another one. Yeah. And yeah, and treated. Yes, pretty good. Yeah, right. And I know. But the difference is, I think, is clear. Right? So much so we feel bad looking at the untreated one. Yeah. Yeah. Okay. Yeah. Right. So I thought, it's better to see it with your own set of eyes instead of hearing about how effective it was. Yeah. So they did test AAV gene therapy via intravenous injection and ICV, the direct injection into the brain. And both of them had the same trend effectively. But we are still following up on the ICV long-term efficacy and safety. This is a safety result. So they did the histopathology, and it was done in a double-blinded way, which means that the person who is checking the safety or looking for concerning signs, they don't know if they are looking at the healthy control or the disease model or the treated. So basically, they did it in an objective way. And they did not find any treatment emergent concerning signs in heart, colon, liver, and kidneys. That's a good thing. So in summary, they did confirm efficacy and safety in mouse and rat model. And we are planning to go for a type B FDA meeting shortly. And Amy's been helping out with the protocol building. And we need to start collaborating closely with you, the community. And the team will be testing in non-human primates, which usually means monkeys. And we'll be checking different doses and long-term safety and efficacy data. So that concludes my presentation, and thank you very much for the collaborators. And I'll take any comments or questions if you have. Or you can simply connect through the organization or me.
Yeah, Yukari, I'll ask you a question.
Sure.
I'll grab Amy's because it's a question for you. I guess, what does it feel like to be a mom to somebody diagnosed with this disease and to have the opportunity and support of your company in pursuing such a rare disease? And how does it feel as a mom and a researcher involved in this to see these results? Like, what's going through your mind? How does this make you feel?
Thank you for the question. I can't deny that my hopes are high, but I'm also realistic. I understand as a scientist that these data might not be necessarily translated into human use. So I'm cautiously optimistic, I would say. Yeah, but sometimes it is hard to separate my personal circumstances from everything else, my professional life, because you can't separate. But I have to stay objective, and I have to have firewall among the different roles that I play. And that's challenging at times. Yeah, thank you.
I think I just want to point out that the UMass study And our natural history study at CHOP was not funded initially by industry. It was funded by Elisa's Corner and AxD, right? And so the community has really been instrumental in supporting the work that has led to these pharmaceutical companies now taking over. And to both Astellas, UMass, and Ionis, on behalf of the patients, thank you so much for investing in Alexander disease, investing in these families. You get a lot of flack from patients and the community asking when and why and faster and how many more patients and all those other things. But, just speaking for everyone, we're incredibly grateful that you chose Alexander disease to work on and have been bringing these things to trial.
Thank you, really.
And that's true. The patient community also helped fund the early studies in the rodent models with the antisense treatment as well. Some of that was NIH funded, but not entirely. So yes, definitely the patient community helped with all the things that we were hearing about today.
And to think too, just one final thing is just to think, this has been within how many years too? I mean, obviously there was a lot going on, right, before the community really, really jumped in and got involved. But think of how much has happened in the past 10 years. And I know 10 years when you're newly diagnosed feels like a lifetime and you feel like you're running out of time and you're just so desperate to cling on to things. But I feel like in the medical world, that is insanely quick how fast we've all come together and how much we continue to band together. And anything the community or anything that the researchers, doctors, anything anyone has asked us to do, we've shown up and we've done it and we've really, really been united as a community. So I'm hopeful that, the people newly diagnosed in the room and then on the phone too are hearing that there is so much happening and it's not today or tomorrow. Trust me, I wish it just as much as everyone else, but it's happening and we just need to do our part to continue to make sure we're staying focused and we're supporting everyone who needs us. So
the rat model itself. Just one more. The rat model itself, which is a terrific, at least in my mind, a terrific preclinical model, and of course, one step along the way, was in part developed from funds derived from patient advocacy groups that we received in the lab.
And thank you for creating.
Yeah, and I guess we're focused on the last 10 years, but to be fair, the journey for you and for Albie started way before the patient community and Elisa's Corner was founded and all those other things. So you've been doing this for way longer, but it's good to hear that even in those days, before Elisa's Corner, before AxD, that it was really patient advocacy that funded some of it.
The first paper was published in 2001, but Albie's paper in 1996 showing the overexpression of human wild-type GFAP generated Rosenthal fibers in mice. I think that's what led to looking into patients and finding out. Yep. Yeah. Yeah. Yes. Yes. But that was the first clue that it was GFAP was the culprit in Alexander disease.
And I don't want to take the mic, but I want to pull it together. Yukari put up a really important graph, and I think Phil asked the question about the 2.5 billion. That timeline, all that money, that includes this timeline and this research, all the way back to primary basic findings of what is this silly thing in biology? There's something happening. Is it related to something that matters in people? And is that a bad thing that happens, a disease in people that leads to a discovery or leads to a treatment that's approved? This timeline, that $2.5 billion includes all that research that Albie, Tracy, and others have been doing for decades. That's included.
And can I mention the reason why I could convince our company, which is Astellas Pharma, was because there is that model already, established, and pathophysiology was already established thanks to natural history study. So really, everybody is contributing to, make this happen. Yeah, thank you.
Yeah, thanks to everybody.
Did you want?
Is that up next?
And Jen, it's 4:30. Yeah. So I want to give a time check for people in case anybody's got to like catch a flight or something.
I've got a flight to catch, so I've got probably like 15, 20 minutes left to go, but let's just.
They're not going to kick us out of the room. Like, we can have this room. I know that.
Yeah. Yep.
How are you for timing?
I'm here through tomorrow.
Oh.
I'll see you at the bar later.
I'm out around 5. Right? I'm hoping to get a ride for someone. Yeah, we're driving.
Jenny, can I have one note from you?
Oh, yeah.
Oh my gosh. Oh, that's great.
I want to motivate our CEO.
I love that.
So if everybody could
put something about
put
funding
something.
Yeah.
I tell you,
something there about funding.
I tell you, bring pizza at the airport. That's not going to make it on your like 36-hour flight.
So I have a question. So when it comes to, well, I don't know if there's people still on the line. What's the sort of sums that you receive, your initial, sponsors by your initial advocates, like you said, Elise's Corner, like, yeah, obviously getting funding from these big donors is great, but I don't know, I'm trying to see, like, what's the value, like, to start these research? Is it like studies? Is it millions or like hundreds of thousands? Like, what's the number? I just have no idea.
I mean, any bit helps, right? Every bit helps. I think that, there are some projects that can be done, like there are pilot projects that are $10,000 to $20,000. There's pilot projects that are $50,000. Then there's more complicated projects that get into the $200,000 to $500,000. And then when you get into these big trials, that's millions, right? But I would say a meaningful amount to get a pilot project done might be $20,000. What do you think?
It depends on, if you're talking about animal studies, it adds up very quickly.
Yeah.
Because you're talking animal housing, animal treatments, staff. It's a lot of work. And so, I think we've tried to put estimates together, and it's hard to put a number on because you have to you can't just hire somebody for one specific study.
Yeah.
But, to do a thorough test, it's going to cost, anyway, to just test something, I would put it in the range of $150,000.
Okay.
Yeah, on average, like the average advantage puts out to do a well-designed efficacy test, preclinical model, and animal model, like a mouse or rat, it's $200,000 to $300,000.
Yeah.
Beginning to end, like that is generally what they look to fund over a number of years to do to get an actual answer. You're going to test that more than once likely.
And that's, I think that is one of the most important things that we from NAXD has done is we've stood up a scientific advisory committee in which you're on the board, right? You're not on the board? Oh, I thought you were. Well, maybe next year. But Yukari's on the board, Dr. Waldman's on the board. I mean, we have a whole panel of experts who are helping guide us in terms of what the best projects are for us to invest in. So that way we're not just blindly throwing money at every different direction, but rather we're trying to take a very systematic approach to saying what's our biggest bang for our buck, what are some really low-hanging fruit that's going to get us some good results versus what's better for long-term investments too. So,
So in addition to the study, if you want to move forward the clinical study, you need to make the drugs for injection. So for example, ASOs are relatively cheap, but in case of AAV, this is a virus vector. You need a lot of funds, at least million. It's easily up to $1 million to start a clinical trial. So that's another, investment you need.
Very happy to talk more about the cost. It's such an important question.
Yeah, of course, it depends on your starting point. When we were working with Ionis, they had done all of the PK work, so pharmacokinetics and pharmacodynamics, and so we didn't have to do any of that. And so, it's hard to know final costs, but, just for basic experiments, it's still quite pricey.
people online? Yes, there are. Okay, so I'll speak into the microphone. It's Jason Child, and I'll move up a little closer. Okay, so the update I'm going to be primarily giving is really intended to get you all to say, yes, I will help. So that's where we're going to end. So I'll start with that. Yes, I will help. And I'll show you a timeline. First, my disclosures, because I know all the doctors and scientists and clinicians are giving disclosures. I have no financial motivation. And like Thomas said, I'm just a dad trying to do the right thing. And the nice thing is I don't think I've spoken to anybody, including some new people that I've met for the first time, as well as people online and in person, that have said, I'm not going to help. So this is a very easy discussion for me to have, because I am taking this as an opportunity to bring an opportunity for you to help out in supporting the community and supporting our all future drug development and research, all of these great things that we're reading about and hearing about at a much more increased pace now, more so than ever before, and definitely more than 10 years ago. We're hearing about all these new developments, and ultimately the search for a therapy, a treatment is there, and it requires approval. Why I'm involved is because in order to get something approved, someone needs to know what it's like to be an Alexander disease patient, family member, caregiver, or participant, and collaborator, researcher, community member, physician. So with all of that, very quick update. We have, we are hosting an externally led patient-focused drug development meeting with a division of the U.S. government that is involved in drug approval. I am allowed to? I don't even know. As a Canadian, I don't even know if I'm going to be able to get out of this country. I might be kept. Anyhow, the slides that I showed on the AxD community engagement are here. They're also available online on the website, etc. The key to an insightful, robust, and informative externally led PFDD meeting is active community outreach to ensure a representative group of patient perspectives. I read that because those are their words. And I stress, and this is how I got involved, was that I was communicating with the AxD board, and they said, okay, well, we have this externally led PFDD meeting. What do we need to do to get involved? And I made a point to say we have such a small community that it is far more important for us in the Alexander disease community to have a much higher representation and participation rate because of the size, effectively the size. We need 25 families to participate in our meeting. And that can be pre-recordings, it can be live attendance, and it can be submissions, it can be surveys, and everything else. So we need 25. I want to make sure that we're drawing from a pool of 50.
And that's outside of the U.S., inside the U.S. It doesn't matter where, right?
I'm Canadian, and I'm participating. The question I asked our friend from Italy, who gave that great presentation and then had to unfortunately leave, was how do we connect to that community? So hopefully it's through discussions like this and questions like this that say, oh, hey, Jason, you asked me about how I connect to this group. Here's 5 more families. Or hey, Dr. So-and-so, we talked about research and collaboration. Here's 4 new patients. And if everybody knows one more person that's not here, we will get to that number very quickly. We cannot, and this I think is why they put me to lead this, we cannot stand by and let somebody else do this for us. So everybody in this room has to either sign up, raise their hand, or make sure that they have someone sign up for them. Okay, it doesn't mean that anybody's being pressured to participate, but there are enough of us out there. We just need to make sure that they know how to do it, and that's through info@axd.org. Then that you get onto our list, and then we can start communicating properly.
So, Jason, I think people are probably asking, what are we signing up for?
Yeah, so it's even easier than that. I have put up here a 217-day plan. When I wrote this, I don't know what the 217 days are between. I think it's now and January 31st, potentially. The ultimate goal is to get that voice of the patient report written. Here's what's happened since I last did this talk. We have engaged our consulting team, lobbyist team in Washington, D.C. is engaged, the legal team. They are kind of the brains and the coordination behind the whole operation. We have a technology team that is also signed up and on board. We also have 2 medical writers that have come on board as well. So we have now at the AxD level put in all the pieces to put on a production. So now all we need is the content. The content and the meeting structure is fairly prescriptive. There is some flexibility, but it's fairly prescriptive. It's hosted by our community representatives alongside a CDER employee, which is part of the FDA umbrella. And so the 2 of them, along with our consultant, host a mediated discussion. And Rob, you participated in these before, so maybe I'll let you talk about the logistics. But as we understand it, there are certain sections. There are pre-recorded videos that talks about what it's like to have Alexander disease in our lives. So that could be from Olive's perspective. It could be from Yumiko's perspective. It could be from my perspective as a parent. It could be from somebody's grandmother as a caregiver. It could be as a teacher. It could be as a clinician. It could be as anything else. It's how does this affect us day to day, and why is it so important that we are able to pursue these therapies to help make our lives a little bit easier and to chase that the cure of this disease, or at least the treatment of the disease, and how will that greater impact our lives to the positive. And so there will be effectively a group sign-up sheet, and then we will be assigning different responsibilities. We're not at that stage now where I can say, okay, I need you to film your story for 5 minutes, and I need you for 5 minutes. But there will be pre-recorded sessions that tell your story at home, filmed at home with your own families, and that will be 5 to 10 minutes of content that's then pared down. There will be professional support. There will be coaching and guidance, and there is no pressure whatsoever. So this is actually quite fun for somebody like Olive who can't stand behind a microphone and not talk. So what is it? You have a question?
Like you said, from my perspective, do you mean because I experienced Alexander's disease prior to?
Yes. So from the patient's perspective, from the parent's perspective, or from the caregiving or the community perspective. So this is a way that we can highlight the challenges that we have day to day, the constant medications, the constant trips to the doctors, the constant trips, the lack of information, the lack of support, and really the loneliness and the ability for us to have hope. And that again ties into to Thomas's video and that whole characteristic of that. We have a number of participants. When we had the org chart up, I had a good idea, and I think this is how we're going to do it. But everybody in our community needs to be on that chart, and we just need to take off the word board, and we need it to say AxD community, and everybody can get into one of those silos and assign themselves a responsibility. And it's not so much a responsibility in terms of being accountable, it's a responsibility in terms of, hey, I want to participate. I want to help fundraise. I want to help the patient advocacy and the PFDD, which is a real focus of ours right now, or I want to be on the community support side. And if you can put your loved one up on that chart, then you can be part of this community, which is really what we have, and it's what we need. Okay, so very, putting some dates to it. We have submitted as of last week the 3 dates proposed. December 5th, December 11th. I believe it was the 5th, the 17th, and one in January. So there were 3 dates. That's been corrected since this was submitted through, so it's real-time live document. Right now we're at the point where we need to find 25 families, and we have effectively 3 weeks to do that. So I'm 1, 2, 3, 4, 5. Okay, and we can count on the members of the board. We've got a couple online. All right, I feel like I'm hosting an auction here. So this is really important. We started this session today out with, all of having to go out to get a box of Kleenex because there were a lot of tears and there was a lot of sadness, and there is in our day-to-day lives, especially for the newly diagnosed. There's a loneliness. There's a lack of information. You can't look and say, oh, I'm exactly like this person next to me, therefore I know what's coming. That uncertainty. So there is a lot. It started off a little bit sad, and I hope it ends up this ends up with some positivity, because we all work together, do the little things right, big things will happen. It's a cliche, and it's really what we can all sign up for, with all of us pushing the same direction or pulling the same rope, as they say, and we'll be able to make huge strides, and that is the goal. So thank you to Jenny, the AxD board, for bringing me on and bringing me part of this. We are very organized at the procedural level for this PFDD meeting. Now we need the content. And so I will be a lot more vocal in terms of encouraging that. And over the next couple of weeks, there will be requests. Email, I think, is how we're going to communicate in order to get people onto certain lists and to start sharing ideas and timelines with that. That will follow the sheet that you can see up on here. Dr. Waldman has a question.
Are you also looking for families who have lost their loved one to tell their story?
Yeah, we are looking for anybody that has a willingness to participate with sharing Alexander disease stories.
So, because I guess I was worried that families might not be as much on AxD's website anymore if their journey is more in the past.
It's a really good question. One of the key things that Jason talked about was that from the FDA's website is representative. So that's not, representative in like male, female, black, white. It's what matters. It's representative as it relates to the disease. So like in this room we have an adult, you have infant onset, juvenile onset. So like it's what matters. So like as you are analyzing with your consultant what the disease looks like, and Jason and team go down the list of volunteers, it may be one way to think about it. Like if there is a representative part of the community that's not represented with living patients or living family members, or less represented, then there's a really good opportunity to bring somebody in.
Yeah.
Generally, that's the advice you're going to get from a consultant, is kind of start with those that can talk about today's experience, and then if you can't do that, fill it out.
I can assure you all that there was a very detailed survey asking a lot of different questions, trying to be able to segment different things like that. And James has been very helpful in terms of trying to help us with that, and Larry and all that. So we're working the process, and not everyone that submits their names are probably going to be selected either. I think we're really just trying to make sure that we have the most diverse set of people that we possibly can. But we're on it.
Sorry. The way that it's been laid out is that we will have there are allocated times for select video submissions. I think those are the ones that would require the most in advance preparation. There is also a live Q&A where we will have selected families and or representatives from those families, where they will be asked questions kind of ad hoc but almost scripted. And then there will be a live, it's open to the public, so there would be an encouragement for live participation from the community to weigh in on real-time surveys and things of that nature.
And I had heard some things like you don't want to go to the FDA saying, oh, this is a terrible disease. I mean, it is. But you want to go to the FDA with like specifics that they can remember or hold on to. Like, obviously, we want people to tell their own story. We're not telling people what to say. But is there something, how do we get people to tell an impactful thing that might be meaningful to the FDA, right? I mean, like, maybe lumbar punctures aren't so bad, or I don't know. Like, I'm just saying.
We're working with the same people that Maria did, so I think we've got a good.
Yeah, I can tell you so like two things real quick for our time. One, your colleagues from the ALSP community just did this 2 weeks ago. So the Sisters Hope Foundation held a PFDD meeting with the FDA, and I'd encourage you all to reach out to them, watch it. It's available on their website today. So that's from a leukodystrophy perspective.
On the Ionis website?
No, it's on the Sisters Hope Foundation, which is an ALSP foundation. I would define any public engagement with the FDA to help them understand these diseases as successful. What they do with the information next is kind of what you're alluding to. So to take it back, like, do you want to talk about what this is going to get used for?
I will after you.
Yeah, the best advice I can give you is think about if you were all, I think, unified with the shared purpose of advancing knowledge about Alexander disease and using that knowledge to advance treatments and eventually other things like that 4-letter word, cure. The FDA will use this information in its decision-making when those proposals come to the FDA, both clinical trial proposals and applications to approve drugs. So this voice of the patient report is an actual document that goes on the desk of every reviewer of an IND or an NDA, which is the terminology for clinical trials or applications for drugs, and the material that we give them. So they get our drug application, and then they have the voice of the patient report. And they're going to use that to make their decision. Because they can't call you, Phil, and say, hey, what are you thinking about today? But they can go back to the tape, so to speak, and look at the data they see in the application and ask, what do you think Phil would say about this? Does this relate to what was important to Phil and his family? So this really will influence decision-making, and you can see that over and over again in rare disease reviews by the FDA. I hope that's helpful, but like, this is a really serious activity, and usually communities get, without putting pressure on you, but you get one shot. Most communities do not get a second EL PFDD with the FDA. So you get kind of one shot to define what they should be thinking about as they review this.
I think this is something that's so important. I hate that we're running out of time on this, because this is how we let our voices really be heard. You know, I mean, this is the biggest thing that our community has in front of us. Like, this is 100% our number one focus. And I hate that we're running out of time to talk about it. But, you know, Jason, like, I think from my perspective, I don't think it would hurt for us to have, I know we talked a little bit about it on our, you know, we had a community-wide meeting, but I think it would probably be helpful as we continue to go, maybe just having a specific, having another one just strictly focused on this, just so we can talk about this, just the community, so that way we can continue to reinforce the importance of it. Because again, rather than cramming it in 15 minutes before we wrap up, but it's, I mean, it has to be way before.
We need it. Yeah, we're good. Like,
Yeah.
we're good. We're good for the planning. We're good for the recruitment. You guys have all been part of the first stage of recruitment, so thank you. And you're all signed up. So the question for those online was, are we bringing our kids? So it doesn't have to be in person for everybody. And so that we can get through in terms of who goes there. It is a virtual, accessible meeting for all. And I think that's really important as we have such a geographically diverse community and such a small one. It actually works in our favor to be remote. There will be some in-person participation, and I think they like to limit that to either 2 participants or you know, a consultant and a participant. So it will be limited at that point, but there will definitely be some patient involvement all throughout, and recorded and documented as such. In terms of speaking just to Rob's point very quickly, the if I like to think of this as the tiebreaker. So if the FDA is considering a drug that is completely, perfect and should be approved, then it should be approved. If the drug is borderline, then they will go back to certain documents to say why. Like, how important is this to this community? What else does this community have? Is there another one right around the corner? And so when we can talk about 10, 20 years of research having nothing to show for it other than good ideas, but that we've got a community that's come together, that's global, that's organized, that's sophisticated, that's following the rules, that is doing everything right, that's paying attention with science, that's using AI, that's getting research from all four corners of the globe to get us something to hang on to and to look forward to. That's when the FDA says, let's look at this one, right? That's what we want. We want to give them everything they possibly need so that they don't have an excuse to look at another drug because their time is limited. So if we can do that, then we've done our part. Yes. Oh, yeah. For the people online, I think we've had some key departures, so we're just going to pause and take a photo.